A human population can disappear so completely that no name, no language, and almost no recognizable history remains behind. Yet extinction does not always mean erasure. Ancient DNA has shown that fragments of vanished populations can survive inside later humans, preserving evidence of encounters that archaeology alone may never reveal. The unsettling paradox is simple. Some people were lost from history, but not entirely from humanity. Their bodies vanished, while parts of their ancestry continued forward. Ancient DNA revealed a forgotten human lineage, but who were they? For most of human history, the dead left behind almost nothing that could speak clearly about who they were. Bones could survive, sometimes teeth, occasionally a tool or a trace of fire, but the deeper story of ancestry was usually hidden. Then a small fragment from Denisova cave in southern Siberia changed the scale of what scientists thought a fragment could reveal. It did not look like the remains of a lost people. It was simply a piece of a finger bone from an ancient individual, too incomplete to provide the anatomical picture that usually guides the identification of a fossil human. Yet inside that fragment, molecules had survived. When researchers recovered ancient DNA and compared it with genetic material from Neanderthals and modern humans, the result did not fit comfortably into either known group. The sequence pointed toward a population related to Neanderthals, but genetically distinct enough to reveal a separate history. That was the beginning of what we now call the Denisovans. The importance of the discovery was not that one tiny bone suddenly told us everything about an unknown people. It did almost the opposite. It showed how much human history could exist beyond what bones alone had allowed us to see. For generations, the human past had been reconstructed largely from visible evidence: skull shape, teeth, stone tools, and the layers of earth in which remains were found. Ancient DNA added another kind of evidence, one that could preserve relationships even when physical appearance could not be reconstructed. This matters because a population does not need to leave behind a complete skeleton for its ancestry to survive in the scientific record. A few cells trapped inside bone can preserve genetic information long after almost every recognizable feature of a person has disappeared. In Denisova Cave, that information opened a door onto a branch of humanity that had not been clearly identified from anatomy alone. The cave had already contained evidence of ancient human occupation, so the first assumption was not that an unexpected lineage was waiting inside a tiny fragment. That is what makes the discovery so revealing. Science did not begin with a dramatic legend about a forgotten people and then search for proof. It began with a sample, a laboratory result, and a pattern that required explanation. The name Denisovan came later, borrowed from the place where the genetic evidence was first recognized. Even now, that name can give a misleading sense of completeness. It sounds as though we know a clearly defined population with a familiar face, a known homeland, and a detailed culture. We do not. The genetic evidence is strong, but the physical record remains limited, and researchers continue to study how widely Denisovan-related populations were distributed and how diverse they may have been. This uncertainty is not a weakness in the story. It is one of the most important lessons in the study of human origins. A lineage can be real without being fully visible. It can be detected through relationships written in DNA, even when its bones are scarce and its material culture cannot be assigned with certainty. The discovery also reminds us that the beginning of our own species was not unfolding in an empty world. Long before modern humans became the only surviving human population, Eurasia was inhabited by other groups with their own histories. Some of those groups met, separated again, and sometimes had descendants together. Ancient DNA can preserve those contacts in ways that caves and artifacts may not. The finger bone from Denisova Cave therefore became more than a fossil. It became evidence that the human family tree was missing branches, and that some of those branches might still be detectable only through the genetic traces they left behind. If one fragment could reveal an ancestry that had escaped recognition for so long, then the deeper question is how many human populations may have lived, mixed, and disappeared before we ever learned their names. Human evolution is often imagined through complete skulls lined up in museums, each one appearing to represent a clearly defined population. The real archaeological record is much less orderly. Most individuals who ever lived left no fossil at all, and many of those who did are represented by only a tooth, a fragment of jaw, or a small piece of bone. Preservation depends on geology, climate, burial conditions, chemistry, and chance. In warm or acidic environments, bone can decay quickly. In other places, remains may survive but never be found. Even when a fossil is recovered, its shape may not contain enough distinctive information to reveal exactly which population the individual belonged to. This is why the Denisovan story is so important. Their existence was not recognized because archaeologists uncovered a complete skeleton with unfamiliar anatomy. Genetic evidence exposed a relationship that the visible fossil record had not been able to show. A small fragment could carry ancestry even when it carried almost no useful shape. That difference between form and inheritance changed how researchers could think about the human past. Bones record bodies, but DNA can record connections between populations. A skull may reveal features shaped by growth and evolution, yet those features do not always divide ancient groups into simple categories. Closely related populations can share many anatomical traits, and variation within one population can overlap with variation in another. A fragmentary fossil makes that problem even harder. Without enough of the skeleton, researchers may be unable to compare the features that would normally support an identification. Ancient DNA, when it survives and can be recovered reliably, provides another line of evidence. It can show that an individual was more closely related to one ancient population than another, or that populations once exchanged ancestry. This does not make genetics a perfect replacement for archaeology. DNA preservation is uneven, and many ancient remains contain too little usable genetic material. The oldest human history is therefore reconstructed from several incomplete records at once. Fossils, artifacts, sediments, proteins, environments, and genetics. Denisovans became visible because one of those records preserved something the others had not made obvious. That possibility matters far beyond a single cave in Siberia. Human populations could have lived for many generations in regions where preservation was poor, where few excavations have taken place, or where their remains resembled those of related groups closely enough to resist easy classification. Their disappearance from the visible record would not mean that they never existed. It would mean that the evidence available to us is selective. The history of our origins is filtered by what happened to bodies after death and by what later researchers happened to discover. Ancient DNA weakened one quiet assumption that it shaped earlier pictures of human evolution, that the major populations of the past would eventually be recognizable from their bones. The Denisovan discovery showed that a population could be genetically important while remaining physically elusive. It also raised a difficult question about the people encountered by early Homo sapiens as they spread across Eurasia. If some ancient groups left only scattered remains, then the landscape into which our ancestors moved may have contained more human diversity than the fossil record alone suggests. Some populations may have vanished without leaving an obvious anatomical signature, while portions of their ancestry passed into later people. This is one reason the search for human origins cannot begin only with what can be seen in a museum case. The deepest starting point is population history itself, groups forming, separating, moving through different environments, and sometimes meeting again. Fossils preserve rare moments from that process. DNA can sometimes connect those moments across generations. Denisovans emerged from that gap between what was visible and what was inherited, and the gap is still telling us that the human past was probably more crowded, fragmented, and difficult to recover than the surviving bones first allowed us to imagine. For a long time, the history of human origins was reconstructed mainly from things that could be held, measured, and compared. A skull could reveal the shape of a face or brain case. A tooth could suggest age, diet, or biological relationships. Stone tools could show that people had occupied a place, even when their bodies were gone. But every one of these records was incomplete. Ancient DNA introduced a different kind of evidence because it could preserve relationships between individuals and populations. Instead of asking only what an ancient person looked like, researchers could begin asking who shared ancestry with whom, which populations had separated, and whether groups that appeared distinct had later met again. The Denisovan discovery became one of the clearest examples of this shift. A fragment that offered almost no recognizable portrait of its owner could still reveal a genetic history different from both modern humans and the Neanderthals already known from fossils and DNA. That did not create a complete biography of the Denisovans. It created a new problem. If a population could be detected genetically before scientists possessed anything close to a complete skeleton, then the visible fossil record could no longer be treated as a full inventory of ancient humanity. The implications reached directly into the question of where modern humans came from and what kind of world surrounded our ancestors. Homo sapiens did not expand across a landscape occupied only by empty territory. Other human populations had lived across parts of Eurasia, and ancient DNA showed that some were close enough to our ancestors to have children together. Their histories could not always be drawn as separate branches that never touched. Population lines could divide, remain apart for long periods, and later reconnect. Genetic material could cross those boundaries and continue through descendants long after one population ceased to exist as a distinct group. This is especially important for understanding Denisovans because their story is not confined to the few remains associated with them. Traces of Denisovan ancestry survive in some human populations, which means that a vanished population can remain present biologically even when its archaeological record is sparse. Ancient DNA therefore changed the meaning of disappearance. A people could lose their identity through demographic decline, migration, absorption, or other processes without every element of their ancestry vanishing. Researchers must be careful here, because genetic evidence cannot tell us everything. It cannot reconstruct a language from a genome. It cannot reveal what an ancient person believed, what stories were told beside a fire, or how a community understood the landscape around it. Those details require other forms of evidence, and many may never be recoverable. What genetics can do is place limits around what is plausible. It can reveal relationships that were invisible, test whether populations exchanged ancestry, and sometimes show that a simple model of human evolution is too narrow. That is why the beginning of the Denisovan story matters beyond Denisova Cave. It forces us to reconsider the starting picture of humanity itself. Our origins were not produced by one population moving forward alone, while every other human group stood apart. They were shaped within a network of populations that separated, adapted to different regions, sometimes disappeared, and sometimes contributed ancestry to people who came later. The forgotten lineage revealed by ancient DNA was therefore not merely an additional name to place beside Neanderthals and Homo sapiens. It was evidence that the human past contained relationships that fossils had not preserved clearly, and that the history written inside genomes could reach where archaeology remained almost silent. Before we ask what Denisovans looked like, where they lived, or how widely they spread, we first have to accept the quieter lesson their discovery introduced. The beginning of our human story may contain people whose existence survives most clearly not in faces or skeletons, but in patterns of ancestry carried forward through generations. Denisova Cave sits in the Altai Mountains of southern Siberia, in a region where forests, valleys, rivers, and open landscapes have shifted repeatedly with changes in climate. Today, it is known because of ancient DNA, but the cave itself had a much longer history before genetics gave it a new meaning. Archaeological layers inside the cave contain evidence that different human populations used the area at different times. Stone tools, animal remains, sediments, and fragments of human bone accumulated gradually, creating a record that is rich but difficult to read. A cave is not a sealed room in which one group lives continuously for thousands of years. People can arrive, leave, return, and be replaced by others. Sediments can move, and later activity can disturb earlier deposits. For that reason, researchers do not treat every artifact found inside Denisova Cave as belonging automatically to one population. The importance of the site comes from the way several kinds of evidence can be compared. Ancient DNA recovered from human remains has shown the presence of Denisovans and Neanderthals, while genetic evidence from one remarkable individual demonstrated that these populations did not always remain separate. The cave, therefore, represents something more complex than a homeland belonging to a single forgotten people. It was part of a wider landscape through which different human groups moved over long periods of time. That setting matters for the question of human origins, because the Altai region lies within a broad zone connecting different parts of Eurasia. Populations moving through Central and Northern Asia encountered changing environments, mountain corridors, river systems, and ecological boundaries. We cannot reconstruct every journey that took place there, but repeated evidence of ancient occupation shows that the region was used across different periods. Denisova Cave preserves traces from this deep sequence, allowing researchers to place genetic discoveries within physical space. The forgotten lineage revealed by DNA was therefore tied to a real landscape where people sought shelter, processed materials, and left behind fragments of their bodies. Yet caution remains essential. A stone tool does not carry a label naming the population that made it. An animal bone marked by human activity does not reveal whether the person responsible was Denisovan, Neanderthal, or another visitor unless independent evidence connects them. This is why the cave is informative but incomplete. It contains many signs of human presence, but identity must be established carefully rather than assumed. The deeper record also challenges a simple picture in which one human group occupied a territory and was then neatly replaced by another. Genetic evidence points instead toward populations whose histories overlapped and whose boundaries were sometimes crossed through reproduction. One individual from the cave had a Neanderthal mother and a Denisovan father, direct evidence that people from these two ancient populations could meet and have children. That discovery does not tell us whether such encounters were common, peaceful, rare, or socially meaningful. DNA cannot recover those details. What it does show is that the lines separating ancient populations were biological histories, not permanent walls. This matters when we return to the beginning of our own story. Homo sapiens emerged within a world already shaped by other human lineages, and some of those lineages had moved across Eurasia long before modern humans reached the same regions. Denisova Cave offers a rare window into that older world because it preserves both archaeological deposits and genetic evidence from people who would otherwise be almost invisible. The cave does not give us a complete portrait of Denisovans, and it should not be treated as the center of everything they were. Their genetic traces suggest a history extending far beyond the Altai. But the cave gives us a fixed point from which this hidden lineage first became scientifically recognizable. Beneath layers of sediment, human diversity survived not as a complete story, but as scattered evidence that different methods would eventually learn to connect. The small finger bone recovered from Denisova Cave did not arrive with an obvious identity. It belonged to a young individual, and by its appearance alone, it could not reveal the existence of a previously unrecognized human lineage. The decisive evidence came from molecules preserved inside the bone. Researchers first analyzed mitochondrial DNA, the genetic material found in the energy-producing structures of cells and inherited through the maternal line. When that sequence was compared with mitochondrial DNA from modern humans and Neanderthals, it did not fit either pattern as expected. The results suggested that the individual carried a deeply divergent maternal ancestry. That finding was surprising, but mitochondrial DNA represents only a narrow line of inheritance, so it could not by itself describe the full population history of the person. The next step was therefore crucial. Scientists recovered and studied nuclear DNA, which contains genetic information inherited from both parents and offers a much broader view of ancestry. The nuclear genome showed that the individual belonged to a population closely related to Neanderthals, yet distinct from them. This population became known as the Denisovans, after the cave where the evidence had been found. The discovery changed the human family tree because it demonstrated that a major ancient population could be recognized genetically before researchers possessed a complete skull or skeleton that clearly represented it. The genetic relationship was also more complicated than a simple picture of three isolated branches. Denisovans and Neanderthals shared an older common ancestry after their lineage had separated from the lineage leading toward modern humans. Later, those ancient populations did not remain permanently isolated. Genetic studies have shown that they exchanged ancestry with each other, and both also contributed ancestry to some populations of Homo sapiens. The finger bone therefore became important not because it offered a clear image of Denisovan appearance, but because it revealed a hidden relationship among ancient humans. It also showed why the beginning of our own history cannot be reconstructed as a straight sequence in which one type of human replaces another without contact. Populations could divide, remain separated for long periods, move through different regions, and encounter one another again. Their descendants could carry genetic material across boundaries that later disappeared from the archaeological record. Even the word Denisovan must be used carefully. It is a scientific label for a lineage recognized through genetic and fossil evidence, not the name these people used for themselves, and it should not create the impression that every Denisovan-related population across Asia was biologically identical. Later genetic work has indicated that Denisovan ancestry found in living humans is complex and may reflect more than one ancient population related to the lineage first identified in Siberia. The finger bone did not answer all of those questions. It created the framework in which they could be asked. It also demonstrated the limits of what DNA can tell us. A genome can reveal genetic relationships, population splits, and episodes of interbreeding, but it cannot tell us what language the individual spoke, what social rules shaped the community, or what meaning people gave to the cave around them. Those details should not be invented simply because the genetic discovery is remarkable. What can be said with confidence is already extraordinary in a quieter way. A young person lived in ancient Eurasia, died, and left behind a fragment of bone. Long after the community to which that individual belonged had disappeared as a distinct population, enough genetic material survived for researchers to recognize that the human world of the past contained another lineage. That recognition brought the story of human origins back to a deeper starting point. Modern humans did not emerge into a world containing only one other familiar human group. Eurasia held several populations with their own histories, and some of those histories crossed. The Denisovan genome became one of the strongest pieces of evidence for that more intricate past. A fragment that could barely describe a body was able to describe ancestry across enormous stretches of time, revealing that the human family tree had never been as empty or as neatly separated as the surviving bones once seemed to suggest. Once the nuclear genome from the Denisova finger bone became available, the The discovery changed from an unusual genetic signal into evidence for a distinct branch within the ancient human family. The mitochondrial sequence had already suggested something unexpected, but mitochondrial DNA follows only one maternal line and can preserve a history that differs from the wider ancestry of a population. Nuclear DNA offered a broader record. Across the genome, the Denisova individual shared more recent ancestry with Neanderthals than with modern humans, showing that Denisovans and Neanderthals belong to closely related branches that had separated after their deeper ancestral population had diverged from the lineage leading toward Homo sapiens. This corrected the first impression created by the mitochondrial genome. The Denisovan maternal lineage looked unusually deep, but the nuclear genome showed that the population itself was not simply an ancient branch standing outside both Neanderthals and modern humans. It was more closely connected to Neanderthals. That relationship gave scientists a framework for recognizing Denisovans as a population history rather than treating the finger bone as an isolated genetic curiosity. The name came from Denisova Cave, but the name should not be mistaken for a complete identity. Researchers did not suddenly know what these people called themselves, what languages they spoke, or whether populations related to the individual in Siberia shared one culture across a vast region. Genetics can identify ancestry and biological relationships, but it cannot supply missing social worlds. The biological picture remained incomplete because one genome cannot represent every person who belonged to a widespread ancient lineage. Later evidence made that limitation important. Genetic traces in living people and additional ancient remains suggested that Denisovan-related populations were not confined to a single Siberian cave, and that their history included geographic and genetic diversity. The first genome provided a starting point, not a finished portrait. It established that another human population had existed alongside Neanderthals and the ancestors of modern humans. It also revealed that these populations could not be understood as sealed compartments. Genetic evidence showed contacts among them, meaning that ancestry sometimes moved from one population into another through reproduction. This makes the idea of a simple family tree useful only up to a point. A tree can show that populations share ancestors and later separate, but human population history also contains reconnections. Branches that had diverged could meet again. When they did, parts of one lineage could continue inside another, long after the original population ceased to exist as a distinct group. Denisovans became one of the strongest examples of this process. Their importance to human origins rests on more than the discovery of another ancient population. They changed the scale at which disappearance could be understood. A lineage could become almost invisible in the fossil record while remaining detectable through DNA, and its genetic contribution could survive far from the place where the lineage was first recognized. This means that the beginning of our own species cannot be separated from the populations already living across Eurasia. Homo sapiens was one branch within a broader human landscape, and when some of our ancestors moved into regions occupied by other humans, those encounters sometimes left descendants. The Denisovan genome turned that possibility into something that could be tested with biological evidence. Yet it also created a restraint. A genome can show relatedness without showing a face. It can reveal shared ancestry without revealing a memory. It can demonstrate contact without explaining what that contact meant to the people involved. The more powerful ancient DNA became, the more important it became to distinguish what the molecules demonstrated from what we might be tempted to imagine. By the end of the first Denisovan genetic studies, science had gained a new branch of human history, but not a complete people. What survived was a pattern of ancestry strong enough to prove that the human past contained a population that had remained hidden in plain sight, waiting inside a fragment of bone for tools capable of recognizing it. When ancient DNA began to place Denisovans beside Neanderthals, The discovery did not create a simple new branch that could be added to a finished family tree. It revealed that the tree itself was only an approximation. Genetic evidence shows that Denisovans and Neanderthals shared a closer common ancestry with each other than either did with modern humans, meaning that their populations were part of a broader Eurasian history that had already separated from the lineage leading toward Homo sapiens. After that deeper separation, the ancestors of Neanderthals and Denisovans also divided from one another. Over time, their descendants occupied different regions, experienced different population histories, and accumulated genetic differences. This is why Denisovans are not simply another name for Eastern Neanderthals. They were closely related, but the genomes preserve enough distinction to show that they followed separate histories for long periods. The comparison becomes easier to understand if we imagine ancestry as a series of populations rather than a parade of species appearing one after another. A population can divide when groups become geographically or demographically separated. Once contact becomes limited, genetic differences can gradually accumulate. If those groups later meet again, however, separation does not guarantee permanent isolation. Denisovans and Neanderthals appear to have done exactly this. Their lineages diverged, yet genetic evidence shows that they later exchanged ancestry. One individual from Denisova Cave had a Neanderthal mother and a Denisovan father, giving direct evidence that the boundary between these populations could be crossed. That single person cannot tell us how frequent such encounters were across all of Eurasia, but it demonstrates that the populations were not completely reproductively isolated. This matters for the origins of modern humans, because Homo sapiens later became part of the same network. Some of our ancestors encountered Neanderthals, others encountered Denisovan-related populations, and portions of that ancestry continued into later generations. Human evolution, therefore, becomes harder to describe as a clean succession of separate forms. The beginning of our own species unfolded while other human populations were already living across Eurasia, and those populations had histories that were connected to one another before modern humans entered many of the same regions. The terms Neanderthal, Denisovan, and Homo sapiens remain useful because they identify major population lineages, but the genetic record warns us not to imagine them as sealed biological boxes. Each label gathers together people who lived across large areas and long periods, and those populations themselves could contain substantial internal variation. Denisovans may be especially important in this respect because the evidence for them comes from geographically scattered genetic and fossil clues rather than from a large collection of complete skeletons. The lineage, first recognized in Siberia, may have included populations that had been separated from one another for considerable periods before some of them met ancestors of living people. Researchers continue to refine those relationships as new genomes and remains are studied. What is secure is the broader pattern. Denisovans belonged close to Neanderthals in the human family, but they were not the same population, and their ancestry followed its own path. That path also reminds us why a branching diagram must be read cautiously. A line splitting into two on paper suggests a permanent division, yet ancient genomes preserve later crossings between those lines. Population history contains both separation and reconnection. The forgotten lineage revealed by DNA is therefore not important because it adds one more isolated figure to prehistoric humanity. It shows that our deeper ancestry developed within a changing community of human populations whose boundaries could open and close across time. Before we can understand where Denisovans lived or how far their descendants spread, we first have to see them in this relational way, not as a mysterious species standing alone, but as one branch of a wider human history that repeatedly divided and met again, leaving genetic traces that survived long after many of the populations themselves had disappeared from view. For much of the 20th century, Human evolution was often described as a sequence of branches that separated and then continued independently. Ancient DNA has made that picture more complicated. Denisovans and Neanderthals were distinct populations with their own histories, yet genetic evidence shows that their separation was never absolute. At different times, individuals from these populations met, had children, and passed ancestry across boundaries that had already existed for many generations. One of the clearest examples comes from Denisova Cave, where researchers identified the remains of an individual whose mother was Neanderthal and whose father was Denisovan. That discovery provided direct biological evidence that two ancient populations, already distinguishable in their genomes, could still reproduce with one another. It did not reveal how often such encounters occurred, what circumstances brought the parents together, or how their community understood such relationships. Those details remain beyond the evidence. What the genome does show is enough to change the structure of the story. Population separation can create genetic differences without producing permanent isolation. Geography may divide groups for long periods, but movement can bring them back into contact. When that happens, genes can move from one population into another and remain there long after the original encounter has been forgotten. The same principle became important when Homo sapiens spread into Eurasia. Modern humans did not encounter a world in which every earlier human population had already disappeared. Some of our ancestors met Neanderthals, and others met Denisovan-related populations. Genetic traces of those encounters survived in later descendants. This means that the ancestry of living people cannot always be represented as a single line moving cleanly from one ancient population to another. It is better understood as a history of populations that divided, expanded, contracted, and sometimes reconnected. That pattern reaches back toward the beginning of our species, because the early history of Homo sapiens developed alongside other human lineages, rather than in complete biological isolation from them. Denisovans make this especially clear. Their genetic relationship with Neanderthals shows that they shared a deeper ancestral population, while later DNA evidence shows that the two lineages exchanged ancestry after they had diverged. Additional genetic signals suggest that Denisovan-related populations also interacted with ancestors of some modern human groups. Each of these events complicates the idea that a population must either remain separate forever or disappear completely. A lineage can lose its independent existence while portions of its ancestry continue inside another population. That is why the disappearance of Denisovans cannot be understood only by asking when the last Denisovan individual died. The more useful question is what happened to their ancestry as populations changed. Some genetic material was lost over generations, some remained. Some may have been carried far from the places where Denisovan fossils are known. Natural selection, chance, population size, and later migrations all affected which fragments survived. Researchers can detect some of those fragments today, but they must be careful not to turn genetic signals into stories that the evidence cannot support. A segment of inherited DNA can show that ancestry once crossed between populations. It cannot tell us whether the people involved saw one another as strangers, relatives, rivals, or members of the same social world. It cannot tell us whether an encounter lasted a day or reflected repeated contact over many generations unless broader genetic patterns support that conclusion. The value of ancient DNA lies precisely in keeping these levels of evidence separate. It allows scientists to identify biological connections without pretending to recover lost thoughts or cultures. Seen in this way, Denisovans do not represent an isolated mystery standing outside human history. They reveal something more fundamental about how human populations have always changed. Separation can be real without being final. Distinct ancestry can persist even when populations meet again. Human evolution, therefore, contains both branching and merging. and the forgotten lineage uncovered through DNA survived, partly because some of those ancient boundaries were crossed. The familiar image of human evolution as a tree remains useful because it shows that populations can share ancestors and later diverge. But ancient DNA has revealed that the history of those branches was not always permanent separation. Denisovans make this especially clear. Their genomes show a close relationship with Neanderthals, yet both lineages also exchanged ancestry with other human populations after they had already become distinct. Instead of imagining human evolution as a set of branches moving forever away from one another, it is often more accurate to think of a network in which populations separated, moved into different regions, changed over generations, and sometimes reconnected. Those reconnections could leave descendants, and the descendants could carry small pieces of ancestry into populations that survived much longer. This is why the discovery of Denisovans changed more than the list of known ancient humans. It changed the geometry of the human past. A simple tree suggests that once two populations divide, their histories continue independently. Genetic evidence shows that reality could be less orderly. Neanderthals and Denisovans had distinct population histories, yet individuals from those groups sometimes had children together. Homo sapiens later entered the same broader Eurasian world and also exchanged ancestry with both Neanderthals and Denisovan-related populations. The result was not a clean replacement of one human type by another, but a changing pattern of contact and inheritance. None of this means that the populations were biologically identical. Genetic differences remained meaningful, and long periods of separation allowed populations to develop their own histories. The important point is that difference did not always create an absolute barrier. When geography shifted, populations moved, or territories overlapped, previously separated groups could meet again. The genetic consequences of those meetings could survive long after the people themselves disappeared from the archaeological record. This brings the question of human origins back to a more complicated starting point. The ancestors of living humans were not moving through a world of empty landscapes and isolated evolutionary experiments. They entered regions where other human populations had already lived for long periods, and some of those populations were themselves connected by earlier episodes of contact. The Denisovan lineage therefore sits inside a web of relationships rather than at the end of a lonely branch. Even the ancestry found in living humans reflects this network. Some populations today carry Denisovan-derived genetic segments, while others carry little or none. The pattern does not mean that Denisovans survived unchanged into the present. It means that ancestry can outlive population identity. Over many generations, migration, population growth, natural selection, and random inheritance can preserve some genetic fragments while others disappear. A population may cease to exist as a recognizable group, yet parts of its biological history can continue inside people who belong to entirely different populations later. This makes the word extinction more complicated when applied to ancient humans. A lineage can disappear demographically while still contributing ancestry to descendants outside that lineage. It also means that identifying one ancient genome does not reveal a complete population. Denisovans themselves may have included groups that had been separated from one another across different parts of Asia for long periods. Genetic evidence from living people suggests a more complicated internal history than a single small population confined to Siberia. Researchers continue to investigate how many Denisovan-related populations existed, where they lived, and how often they encountered Homo sapiens. The evidence does not support turning every genetic difference into a new species or inventing unknown civilizations around incomplete data. What it supports is a quieter and more important conclusion. Human evolution was structured by repeated separation and reconnection. The boundaries between populations were real enough to leave distinct genetic patterns, but porous enough that ancestry sometimes crossed them. Denisovans help reveal this because their history is preserved less through abundant skeletons than through relationships written into genomes. Their existence reminds us that the beginning of humanity cannot be understood only by asking which population replaced which other population. We also have to ask who met whom, which lineages mixed, which fragments of ancestry survived, and which populations vanished while leaving biological traces behind. Once human evolution is seen as a network rather than a simple tree, the forgotten lineage discovered through ancient DNA no longer appears as an isolated exception. It becomes evidence that the human past itself was built from connections that were repeatedly lost, restored, and carried forward in ways that bones alone could never fully reveal. The scarcity of Denisovan remains is one of the strangest features of their story. Their genetic importance is clear, yet for years the physical evidence associated with them consisted of only a handful of small fragments, teeth, and pieces of jaw. That imbalance can make the Denisovans seem almost unreal, as if a major human lineage appeared first as a pattern in DNA and only later began to acquire a body. The reason is not that Denisovans were necessarily rare in their own time. The fossil record is shaped by preservation, discovery, and identification, and each of those filters can remove enormous amounts of evidence. Most human bodies never fossilize. Bone is broken down by water, soil chemistry, temperature, animals, microorganisms, and geological movement. Even when a fragment survives, it may remain buried in a place that has never been excavated. A surviving bone can also be found without being recognized for what it is. Denisovans illustrate that last problem especially well, because many of their known remains do not carry a single anatomical feature that immediately identifies them as Denisovan. Molecular methods have therefore been unusually important. DNA can connect a fragment to the Denisovan lineage when enough genetic material survives, while ancient proteins can sometimes provide biological information when DNA has decayed too far. This has gradually expanded the physical record beyond Denisova Cave. A lower jaw from the Tibetan Plateau was linked to Denisovans through ancient proteins, supporting the idea that Denisovan-related populations lived far beyond southern Siberia. More recently, molecular evidence has also linked additional remains from Eastern Asia to the Denisovan lineage, including a jaw from the area around Taiwan and a large archaic human skull from northeastern China. These discoveries have begun to give the lineage a more visible anatomical presence, but the overall record remains sparse compared with the much larger collection of Neanderthal fossils. That difference should not be converted into a claim about how many Denisovans once existed. A large population can leave a poor fossil record, while a smaller population can appear archaeologically prominent if preservation and excavation happen to favor it. Geography also matters. Denisovan ancestry in living humans suggests that populations related to the lineage, first identified in Siberia, once occupied a broad area of eastern Eurasia. Many of those regions are humid, mountainous, tropical, coastal, or otherwise difficult environments for long-term preservation and systematic recovery of ancient DNA. The places most likely to preserve molecules are not necessarily the places where the greatest numbers of people once lived. This creates a deep distortion in our view of human origins. We see the past through the rare environments that preserved it, not through an equal sample of every landscape our ancestors and their relatives inhabited. Denisovans may therefore appear centered on caves and cold regions, partly because those locations have been unusually generous to archaeology and molecular preservation. The growing fossil record also teaches another lesson. Identification is not always immediate. A jaw or skull can be discovered years before techniques become available to test whether its proteins or DNA connect it with a known lineage. Some fossils already stored in collections may eventually be reassessed as new methods improve, while other remains may never yield molecules strong enough for confident assignment. Anatomy can help, especially as more securely identified Denisovan fossils become available for comparison. But researchers must avoid creating a fixed Denisovan appearance from too little material. Individuals vary, populations change over time, and a lineage spread across a wide geographic range may not have looked identical everywhere. The forgotten human lineage revealed by ancient DNA is therefore becoming less invisible without becoming simple. Each new bone gives the genetic signal more physical context, but it also widens the questions. The few remains do not show a people confined to one cave. They point toward populations distributed across very different environments, some separated by great distances and perhaps by long stretches of time. At the beginning of our own wider human story, this matters because the absence of abundant fossils cannot be treated as the absence of human diversity. Denisovans remind us that entire populations can pass through the archaeological record almost silently, leaving only scattered fragments until new methods teach us how to recognize what was there. For a lineage first recognized through DNA, the physical record of Denisovans has grown in an unusual order. Instead of beginning with a complete skull and then adding genetic evidence, researchers often began with fragments whose identity could not be established by shape alone. Teeth became especially important because dental tissues are dense and can survive when more fragile bones disappear. Several teeth from Denisova Cave have been linked to Denisovans through genetic evidence, and they show that the people represented there were not known only from the famous finger bone. Some of these teeth are large and preserve features that can be compared with other archaic humans, but size or shape alone cannot safely identify every similar tooth found elsewhere as Denisovan. Closely related populations can share anatomical traits, and individual variation can overlap. This is why molecular identification has become so valuable. When ancient DNA survives, researchers can compare sequences directly with genomes from known Denisovan, Neanderthal, and modern human individuals. When DNA is too degraded, proteins preserved in tooth enamel or bone can sometimes retain enough evolutionary information to help place a fossil within the human family. The Xiahe mandible from the Tibetan Plateau became a major example. DNA was not available in the way it had been from Denisova Cave, but ancient protein analysis showed that the jaw was closely related to Denisovans. The discovery gave scientists a much more substantial piece of anatomy and demonstrated that Denisovan-related people were present far from southern Siberia. Later work at the same cave identified another Denisovan-related fossil through protein evidence, strengthening the connection between this lineage and the high Tibetan plateau. A different kind of example came from the Penghu mandible, recovered from the seafloor near Taiwan. For years, its exact relationship to other archaic humans remained uncertain because morphology alone could not settle the question. Ancient protein analysis later linked it to a Denisovan male, providing another molecular point far to the southeast of the place where the lineage was first recognized. These findings do not mean that every robust jaw or unusually large tooth in Asia should now be called Denisovan. That would simply replace one uncertainty with another. Molecular evidence is strongest when it can connect a specimen directly to an established genetic lineage, while anatomy becomes more informative as the number of securely identified fossils increases. There are also cases where researchers can make only a probable assignment. A molar found in Laos, for example, has anatomical and contextual evidence consistent with Denisovan affinities, but the absence of decisive genetic identification requires caution. That distinction matters because the search for human origins depends as much on knowing what remains uncertain as on recognizing what has become clear. Each securely identified tooth, jaw, or bone fragment gives researchers another point of comparison. Gradually, the lineage that first appeared as an unexpected genome begins to acquire a physical record. Yet even this growing record cannot tell us that Denisovans everywhere had one fixed appearance. Populations related to Denisovans may have occupied different environments across large parts of Asia and may have been separated from one another for long periods. Their bodies could therefore have varied across geography and time, just as populations within other human lineages varied. The beginning of our own broader human story becomes clearer through this careful combination of evidence. Fossils provide anatomy and place, archaeological layers provide context, DNA reveals close genetic relationships, ancient proteins can extend identification into remains where DNA has not survived. None of these methods alone reconstructs a lost people in full, but together they allow scientists to move from an almost invisible lineage toward a more grounded picture. Denisovans are no longer represented only by a mysterious genetic sequence from one cave. They are increasingly connected to real jaws, teeth, fragments, and landscapes across Asia, while every new identification also reminds us how much of their physical history remains missing. The more Denisovan fossils researchers identify, the more tempting it becomes to imagine that a complete picture of these people is finally emerging. Yet the strongest scientific approach is to recognize how much still cannot be reconstructed. Ancient DNA can show relationships between individuals and populations. Teeth and jaws can reveal aspects of anatomy. Archaeological deposits can establish that humans occupied particular places at particular times. None of these records, however, gives us direct access to an entire way of life. We do not know what Denisovans called themselves, what languages they spoke, how their communities were organized, or what stories they passed between generations. We cannot safely assign every tool found near a Denisovan fossil to Denisovans unless the archaeological context makes that connection convincing. Nor can we build a single Denisovan face from a few jaws, teeth, and fragments scattered across enormous distances. The lineage first recognized in Siberia may have included populations that differed from one another after long periods of geographic separation. A person living on the Tibetan plateau could have belonged to a population with a different local history from people living much farther east or north. The same caution applies to behavior. The presence of humans in a difficult environment tells us that they were capable of surviving there, but it does not reveal exactly how they organized food sharing, family relationships, ritual practices, or communication. Archaeology can sometimes provide evidence about technology, diet, mobility, or repeated use of a site, but connecting those patterns to a specific human population requires careful support. This distinction matters because prehistoric humans are easily turned into characters. Once a lineage has a name, it is natural to imagine a familiar society behind it. Denisovans can then become hunters, travelers, mountain specialists, or mysterious inhabitants of Asia in stories that move far beyond what the evidence actually establishes. Scientific reconstruction has to move more slowly. The beginning of our own human story is already complex enough without adding invented certainty. What can be said is substantial. Denisovans were a real human lineage, closely related to Neanderthals. Their genetic history was distinct. Populations related to them occupied more than one region of Asia. They encountered other human groups, including ancestors of some modern humans, and some of their ancestry survived in later populations. Fossils and molecular evidence are gradually expanding the geographic and anatomical record. But a genome does not contain a culture in readable form. It cannot tell us whether a group had a particular myth, burial tradition, political structure, or sense of identity. Even physical reconstruction remains limited. A jaw can tell us about the size and shape of the lower face, and teeth can provide valuable comparisons. But these elements do not reveal skin appearance, hairstyle, expression, or the full proportions of the body. Some biological traits may eventually be studied through genetic evidence, yet such interpretations require strong support and should not be extended beyond what specific variants can reliably indicate. The absence of certainty is therefore not an empty space that needs to be filled with imagination. It is evidence about the limits of preservation. Every ancient population reaches us through a damaged archive. For Denisovans, that archive is especially uneven, unusually rich in genetic relationships, but still poor in complete skeletons and securely associated cultural material. This imbalance is one reason their story feels so different from that of better-known ancient humans. We know they existed with confidence, yet we cannot stand before a reconstructed Denisovan individual and claim to see the population as it truly was. That gap also protects the central question from being answered too quickly. Ancient DNA revealed a forgotten lineage, but knowing that a lineage existed is not the same as knowing the people who belong to it. The closer science moves toward them, the clearer the boundary becomes between what can be recovered and what time may have erased permanently. The name Denisovan comes from a cave in the Altai Mountains, but the people connected to that name were not confined to the place where science first recognized them. For years, this created a strange imbalance in the evidence. The clearest ancient genomes came from southern Siberia, while genetic traces in living people pointed toward a much wider history across eastern Eurasia. That wider picture became stronger when researchers identified Denisovan remains on the Tibetan plateau. A lower jaw from Baishia Karst cave was linked to the Denisovan lineage through ancient protein analysis, and later work at the same site recovered Denisovan mitochondrial DNA from cave sediments, as well as another Denisovan-related bone. Together, these findings showed that people connected to the Denisovan lineage had occupied a high-altitude environment far from Denisova Cave. The importance of this evidence is not simply geographic. It changes the scale of the question. If Denisovan-related populations lived both in the Altai and on the Tibetan plateau, then the lineage cannot be understood as a small local group whose history unfolded around one Siberian cave. The genetic record also points beyond the places where securely identified fossils have been found. Denisovan ancestry survives today in varying amounts among populations in parts of Asia and Oceania. That pattern does not provide a precise map of every place Denisovans once lived, because ancestry can move when later human populations migrate. It does, however, show that encounters between Homo sapiens and Denisovan-related groups occurred somewhere along routes followed by the ancestors of later populations. In island Southeast Asia and the Pacific, the genetic signal is important because confirmed Denisovan fossils remain scarce or absent in many regions where Denisovan ancestry is now detected. Once again, genes preserve a relationship that the visible fossil record has not yet matched. The beginning of our wider human history therefore becomes inseparable from geography. As Homo sapiens expanded through Eurasia, our ancestors were moving across landscapes already occupied or previously occupied by other human populations. Mountains, plateaus, forests, river valleys, coastlines, and changing climate zones would have influenced where groups could move and where they might encounter one another. We should not imagine Denisovans as a single population spread evenly across all of Asia. A geographic range that broad would likely have contained populations separated by distance and environment, and genetic research supports the idea that Denisovan ancestry itself reflects considerable population structure. Some Denisovan-related groups may have been more closely related to the individuals known from Siberia than others were. This is one reason the label Denisovan must remain flexible. It identifies a major branch of ancient humanity, but it may gather together populations whose histories had already diverged from one another before they encountered modern humans. The Tibetan evidence gives this idea a physical foundation. It places Denisovan-related people in a setting very different from the Altai and shows that their history reached into environments once thought difficult for ancient human occupation. Even here, caution is necessary. Finding Denisovan evidence on the plateau does not tell us that every archaeological site in the region belonged to Denisovans, nor does it prove exactly how they adapted biologically or culturally at every moment. What it establishes is presence. The same discipline is needed when interpreting genetic traces farther south and east. DNA can reveal contact and shared ancestry, but it cannot by itself identify the exact village, valley, or generation in which an encounter occurred. Still, the direction of the evidence is clear. The forgotten lineage first detected in one Siberian cave was part of a larger human landscape. Denisova Cave was the place where science recognized them, not necessarily the center of their world. Once that distinction is made, the question of who the Denisovans were becomes inseparable from another question. How many Denisovan-related populations may have lived across Asia before some of their ancestry entered the people who came later? The Tibetan Plateau changes the Denisovan story because it places this lineage in a landscape very different from the Siberian cave where their DNA was first recognized. Baishia Karst Cave lies high on the northeastern edge of the plateau, an environment marked by cold temperatures, thin air, strong seasonal change, and ecological conditions that would have challenged any human population living there for long periods. The first major clue came from a partial lower jaw known as the Shie mandible. It had been discovered decades earlier, but its biological identity remained uncertain until researchers analyzed ancient proteins preserved in the fossil. Those proteins linked the individual closely to the Denisovan lineage. This was important for two reasons. First, it provided a substantial piece of anatomy from far outside the Altai Mountains. Second, it showed that Denisovan-related people had reached a high-altitude region long before the lineage was recognized through genetics. Later work at Baishia Karst Cave strengthened the connection. Researchers recovered Denisovan mitochondrial DNA directly from cave sediments, showing that genetic traces can survive even when no human bone is present in a particular layer. Additional molecular work identified a hominin rib from the site as Denisovan-related, extending the physical evidence and showing that the cave was used by Denisovan populations across widely separated periods. These discoveries do not tell us that the same community occupied the cave continuously. Archaeological layers represent long spans of time, and human presence can be intermittent. Groups may arrive, leave, and return, while other populations may use nearby landscapes without ever entering the cave. What the evidence supports is repeated Denisovan presence in this high-altitude region, not a single unbroken settlement. That distinction matters, because prehistoric life cannot be reconstructed as though one fossil represents thousands of years of behavior. Even so, the Tibetan evidence changes how we think about the geographic range of Denisovan-related populations. The lineage was capable of existing in environments very different from the forests and valleys surrounding Denisova Cave. At Baishia Karst Cave, animal bones and stone artifacts also provide evidence of hominin activity, and some animal remains show signs of carcass processing. Researchers have argued that Denisovans were likely responsible for much of this activity in layers where their presence is supported by molecular evidence. Yet caution is still necessary. A cut mark cannot identify the person who made it, and a stone tool does not preserve the genome of its maker. The strongest interpretations come from combining archaeology, stratigraphy, proteins, sediment DNA, and securely identified human remains, rather than allowing one form of evidence to stand for the whole story. The plateau also brings the beginning of our own human history back into view. Long before later Homo sapiens populations became established across many parts of Asia, other human lineages had already entered difficult environments and developed ways of surviving there. Denisovans were part of that older human geography. Genetic research in living people has also suggested that some Denisovan-derived variants contributed to later high-altitude adaptation in Tibetan populations, especially around genes involved in responding to low oxygen. But this should not be turned into the claim that Denisovans alone created the biological adaptations of people living on the plateau today. Modern populations have their own long evolutionary histories, and inherited Denisovan ancestry is only one part of that history. The important point is that contact between ancient populations could transmit genetic variants that later became useful under particular environmental conditions. Baishia Karst Cave therefore connects three different kinds of evidence: the bodies of ancient humans, the environments they inhabited, and the genetic legacy that sometimes passed into later people. It gives the forgotten lineage a geographic depth that a single Siberian cave could never provide. Denisovans were not simply a population hidden in one remote corner of Eurasia. At least some Denisovan-related groups reached the Tibetan plateau, returned to or remained connected with that landscape across long periods, and left enough molecular evidence for their presence to be recognized today. The cave does not tell us what they thought about the mountains, or how they understood the cold, the altitude, or the animals around them. It tells us something more limited and more secure. Their history extended into a demanding environment, and the map of ancient humanity was already broad and complex before our own lineage became the only surviving human one. The Denisovan story becomes more difficult and more revealing when we stop thinking of the lineage as a single population living in one kind of place. The evidence now stretches from the Altai Mountains to the Tibetan Plateau, while genetic traces in living people point toward contacts much farther south and east. These clues do not produce a complete map, but they show that Denisovan-related populations were connected to very different environments across eastern Eurasia. Genetic evidence also indicates that ancestors of populations in parts of Southeast Asia and Oceania encountered Denisovan-related groups somewhere along their movement through Asia. The exact locations of every encounter are not known, and the present distribution of Denisovan ancestry cannot simply be projected backward onto an ancient map. Even so, the broad pattern is difficult to explain if Denisovan-related populations were restricted to a small region around Siberia. Their history appears to have extended across a much wider human landscape. This geographic breadth matters because populations separated by mountains, distance, climate, and changing ecosystems do not remain identical forever. Groups related to the Denisovans may have become genetically structured as they lived in different regions over long periods. Modern genomes preserve signs that Homo sapiens did not receive all Denisovan ancestry from one single uniform source. Several genetic studies instead support a more complicated history involving contact with distinct Denisovan-related populations. That does not mean science has identified a separate species for every genetic signal. Population structure can develop within a broader lineage, just as related human populations can have different demographic histories without becoming different species. The safest picture is therefore not of one Denisovan people spreading unchanged across Asia, but of a wider lineage containing populations whose relationships, ranges, and histories varied through time. Denisova Cave preserves evidence from a cold region of southern Siberia. Baishia Karst Cave places Denisovan-related people on the Tibetan Plateau. A molar from northern Laos has been argued to show Denisovan affinities on anatomical grounds, although its identification remains less secure than fossils supported by molecular evidence. Farther south, living populations carry Denisovan ancestry even where no confirmed Denisovan fossil has yet been recovered. These gaps between fossils and genes are reminders that preservation differs sharply between environments. Warm and humid regions are often hostile to ancient DNA, so the absence of a recoverable genome does not demonstrate the absence of a population. At the same time, Genetic ancestry in living people cannot by itself tell us exactly which ancient population occupied a particular cave or island. When the records are read together, however, they reveal that the human world encountered by expanding Homo sapiens was geographically varied and biologically diverse. Our ancestors did not necessarily move through one uniform population of Denisovans. They may have encountered different Denisovan-related groups at different places and times, although the details remain under study. That possibility connects directly to the beginning of our own history. Some ancestry carried by living humans today came from meetings with populations whose names, languages, and social identities are gone. Their genetic contribution survived because descendants carried it forward, not because the original groups remained unchanged. Denisovans therefore illustrate a deeper principle of human evolution. A lineage can spread across different worlds, divide internally, meet other populations, and eventually disappear as a recognizable population, while portions of its ancestry continue elsewhere. The evidence does not allow us to draw every route or identify every Denisovan community. It does allow us to reject the image of an isolated group trapped in a Siberian cave. What emerged from ancient DNA was a lineage with a history broad enough to cross ecological boundaries and complex enough that a single name may conceal several ancient population histories. The farther we follow their traces from the Altai, the less Denisovans look like one lost group, and the more they reveal a wider, fragmented human world that Homo sapiens eventually entered. When Homo sapiens began expanding beyond Africa in a sustained way, they were not entering an empty continent. Eurasia already held human populations with histories that reached far back before the arrival of our own species. Neanderthals occupied large areas of Western Eurasia, while Denisovan-related populations were present farther east and across parts of Asia that are still being reconstructed from fossils and genomes. The movement of Homo sapiens into this world was not a single march made by one population along one route. Fossils, archaeology, and genetics instead point toward repeated movements, some of which left little or no ancestry in later people, while other dispersals contributed much more strongly to populations that survived. This matters because the encounters recorded in modern genomes could only happen after human populations entered the same geographic spaces. As groups of Homo sapiens moved through southwestern Asia and farther across Eurasia, they met people whose ancestors had been separated from ours for a long time. Genetic evidence shows that some of those meetings produced descendants. Neanderthal ancestry entered the ancestors of many populations outside Africa, and Denisovan ancestry entered the ancestors of several later populations in Asia and Oceania. These events did not happen at the beginning of Homo sapiens as a species. They happened after populations of our species had already developed within Africa, and after some groups had begun expanding beyond it. That distinction keeps the story grounded. The origin of Homo sapiens and the later movement of Homo sapiens across Eurasia are related, but they are not the same event. When our ancestors reached new regions, the human landscape they encountered had already been shaped by other lineages. Those lineages had their own histories of separation, movement, and contact long before Homo sapiens arrived. Denisovans and Neanderthals had already diverged from one another, yet genetic evidence shows that they had also exchanged ancestry. Modern humans therefore entered an existing network rather than creating the first connections within it. Geography would have influenced how often different populations met. Mountain ranges, deserts, river corridors, coastlines, and changing climates could open or restrict routes over long periods. But the archaeological record is too incomplete to trace every movement in detail, and ancient DNA survives unevenly across different environments. This is why researchers avoid treating a single map as a final picture of early human expansion. Genetic ancestry found in living populations can reveal that contact occurred, but later migrations can move that ancestry far from the original place of encounter. Denisovan ancestry provides a clear example. Some of the strongest Denisovan genetic signals today occur in populations far from Denisova Cave, showing that the location where a lineage was first identified is not necessarily the location where most encounters with Homo sapiens occurred. The genetic record also suggests that contact with Denisovan-related populations was more complicated than one isolated meeting. Different segments of Denisovan ancestry in living humans point toward contributions from distinct Denisovan-related populations, which means that Homo sapiens may have encountered different branches of that broader lineage at different times and places. The exact routes remain under study, and not every proposed migration path has equal support. What is secure is the broader pattern. Our ancestors did not simply replace every other human population without biological contact. Some encounters left descendants, and those descendants carried pieces of older Eurasian lineages forward. This brings the forgotten Denisovans directly into the history of people living today. Their importance does not depend on imagining a dramatic meeting between strangers in a prehistoric landscape. The evidence is quieter. Somewhere in the movements that carried Homo sapiens across Asia, population boundaries were crossed. Children inherited ancestry from more than one human lineage. Generations later, most details of those encounters disappeared, while fragments of DNA remained. The movement of Homo sapiens into Eurasia therefore marks more than a geographic expansion. It marks the point at which the history of our species became biologically entangled with populations that had already been living beyond Africa, including the Denisovan-related groups whose full range and diversity we are only beginning to understand. Archaeology can show that two human populations lived in the same region, but it cannot always show whether they met or whether their descendants carried ancestry from both groups. Ancient DNA and the genomes of living people can preserve that kind of contact differently. When populations separated for many generations reproduce, their descendants inherit segments of DNA from both sides. Over time, recombination breaks those segments into smaller pieces, while migration, population growth, natural selection, and chance alter how much survives. By comparing patterns across genomes, researchers can identify traces of ancient gene flow. Denisovan ancestry is one of the clearest examples. Some living populations in Asia and Oceania carry genetic segments more closely related to Denisovan genomes than to Neanderthal or other modern human sequences. Those segments show that ancestors of these populations encountered Denisovan-related people and produced descendants together. The evidence does not allow us to watch the meeting or identify the individuals involved. It records the biological consequence rather than the social event. This distinction matters because a genetic signal should not be turned into a prehistoric scene that the evidence cannot support. We do not know what language was spoken, how long populations remained in contact, whether encounters were rare or repeated, or how people understood one another. What DNA can establish is that ancestry crossed population boundaries. The same principle applies to Neanderthal ancestry in many populations outside Africa, but the Denisovan pattern is especially revealing because strong genetic traces are found far from Denisova Cave. This tells us that the cave where science first recognized the lineage was not necessarily the place where important contacts with Homo sapiens occurred. Later, human migrations could also carry Denisovan ancestry away from its original point of entry, so its present distribution cannot be read as a simple map of ancient encounters. Even so, differences among Denisovan-derived segments suggest that the history was not produced by one episode with one uniform Denisovan population. Several studies have found evidence consistent with contributions from genetically distinct Denisovan-related groups. The exact number of contact events remains under research, and it is safer to speak of multiple episodes or sources than to assign a fixed count where models differ. This brings the story back to the beginning of our own wider human history. As Homo sapiens moved through Eurasia, they entered landscapes already occupied by populations that had been evolving separately for long periods. Some disappeared as distinct groups, but disappearance did not erase every biological connection. If descendants were born from mixed ancestry, parts of that history could continue after the original population was gone. Introgression is the term often used for this movement of genetic material from one population into another through interbreeding and inheritance. It does not mean that two populations became one overnight. Gene flow can occur while populations remain largely distinct, and much inherited material can later be lost. What survives today is therefore only a fraction of the ancestry that may once have entered a population. Archaeology and genetics answer different questions here. A stone tool can reveal behavior at a place. A fossil can reveal anatomy and chronology. DNA can reveal biological relationships and ancestry. When these records agree, the picture becomes stronger. When one is missing, the others must not be forced to supply details they cannot contain. Denisovan gene flow is powerful precisely because it demonstrates contact without requiring us to invent the circumstances. Somewhere across ancient Asia, people from different human lineages had descendants whose ancestry continued forward. The locations, communities, and personal stories of those encounters are mostly lost, but the genetic consequences remain measurable. In that sense, DNA records a form of human history that archaeology alone may never recover, not the memory of the meeting itself, but the fact that the boundary between populations was crossed and that part of a forgotten lineage entered the ancestry of people who came much later across generations. The genetic traces left by Denisovan-related populations do not fit comfortably into the idea of one brief encounter between two groups. When researchers compare Denisovan-derived segments across living populations, they find patterns that are difficult to explain with a single episode of contact. Some inherited fragments are more closely related to the Denisovan genome recovered from Siberia, while others appear to come from Denisovan-related populations that had already diverged from that Siberian branch. This does not mean that every difference represents a separate species, nor does it allow science to name each ancient population. It suggests something more cautious and more useful. The people we call Denisovans were probably not one genetically uniform population spread unchanged across Asia. Different groups related to any Hij may have lived far apart, followed separate demographic histories, and encountered Homo sapiens at different times. The ancestors of living populations in Asia and Oceania, therefore, may have received Denisovan ancestry through more than one episode of gene flow. Genetic models can estimate relationships among these signals, but the exact number, location, and timing of every encounter remain subjects of continued research. This uncertainty is important because a model is not a recovered memory of the past. It is a way of asking which population histories best explain the patterns observed in genomes today. Different models can sometimes produce similar genetic outcomes, especially when ancient populations were themselves structured and when later migrations mixed their descendants. The safest conclusion is not that scientists have reconstructed every meeting, but that one simple encounter is unlikely to explain the full diversity of Denisovan ancestry now observed. This brings the story back to the movement of Homo sapiens across Eurasia. Our ancestors were not traveling through a single continuous human landscape. They moved through regions occupied by populations that had their own histories of separation and movement. A Denisovan-related group encountered in one region may not have been genetically identical to another group encountered farther east or south. If contact occurred repeatedly, each episode could contribute different fragments of ancestry to different Homo sapiens populations. Later, Migrations could then redistribute those fragments, making the present-day pattern even more complex. This is why modern geography cannot be treated as a direct photograph of prehistoric contact. People move, populations merge, and ancestry travels. A genetic segment found in one region today may have entered the ancestors of that population somewhere else many generations earlier. The same caution applies to the social meaning of contact. Multiple episodes of gene flow do not tell us whether encounters were friendly, hostile, accidental, or part of long-term coexistence. DNA records inheritance, not intention. It cannot recover the emotions of the people involved or explain the social relationships that surrounded reproduction. What it can show is that ancient population boundaries were crossed more than once in human history, and that some of those crossings left descendants. At the beginning of our wider story, this changes the idea of what expansion means. Homo sapiens did not simply move into Eurasia and replace every population already there without biological exchange. Nor did all earlier populations disappear before our ancestors arrived. Instead, the genetic record preserves a more layered process in which different groups met at different times. Some ancestry crossed between them, and later populations carried those fragments forward. Denisovans are central to this picture because their fossil record remains limited while their genetic legacy is geographically broad and internally varied. The contrast tells us that the people behind that ancestry were likely more diverse and more widely distributed than the first discovery in Siberia suggested. Their encounters with Homo sapiens were probably not one moment that can be placed on a single map. They were part of a longer history of movement, separation, reconnection, and inheritance across Asia. What survives today is not a complete record of those meetings, but a scattered genetic residue showing that the forgotten lineage entered our ancestry through a history more complicated than one crossing between two isolated groups. A population does not have to remain visibly separate for its ancestry to survive. Denisovans demonstrate this in one of the clearest ways ancient genetics has revealed. The people themselves no longer exist as a distinct population that can be identified in the present, yet fragments of DNA inherited from Denisovan-related ancestors remain inside the genomes of some living humans. These fragments are not complete Denisovan genomes preserved intact from the distant past. They are smaller pieces that have been carried through many generations, broken apart and rearranged by recombination each time DNA passed from parents to children. Over long periods, much ancient ancestry can disappear simply through chance, while other segments remain. The Denisovan contribution found today is therefore only a surviving portion of a much larger history of contact. Researchers identify these fragments by comparing genomes from living populations with ancient genomes recovered from Denisovan remains and with sequences from Neanderthals and other humans. Certain stretches of DNA share patterns that are more consistent with ancestry from Denisovan-related populations than with ancestry entirely within Homo sapiens. When many such segments are examined together, they reveal that the forgotten lineage did not disappear without biological descendants. Some ancestors of living people had children with Denisovan-related individuals, and portions of that ancestry continued forward. The distribution is not equal everywhere. Populations in parts of Oceania carry some of the strongest Denisovan ancestry signals, while lower levels and different patterns occur in several populations across Asia. These differences are important because they suggest that the surviving Denisovan contribution cannot be explained simply by one population receiving every once and then spreading unchanged across the world. Human populations moved repeatedly after the original encounters. Some expanded, some became isolated, some mixed with neighboring groups, and others experienced large demographic changes. Each of these processes altered the proportion and location of Denisovan-derived DNA. This means the genomes of living people are not direct maps of where Denisovans once stood. They are records transformed by everything that happened afterward. Even so, they preserve something that bones alone could easily lose. A Denisovan population might vanish from a landscape. Its camps disappear, its bodies decay, and its language leave no recoverable trace. Yet descendants carrying portions of its genome could move far beyond that original region. In this sense, ancestry can travel farther through time than a population name or archaeological tradition. The connection reaches directly into the origins of people living today, but it must be described carefully. Having Denisovan ancestry does not make any modern population partly Denisovan in a simple cultural or biological sense. Every living population has undergone its own long history since those ancient encounters, and the inherited Denisovan segments represent only one component within much larger genomes shaped by many generations of Homo sapiens ancestry. Nor should the surviving fragments be treated as unchanged messages from a lost people. Most genetic material inherited from archaic populations was affected by evolutionary processes after entering modern human populations. Some variants disappeared. Others persisted through chance. In certain cases, particular inherited variants appear to have been favored because they were useful under local environmental conditions. But these cases must be evaluated individually rather than used to claim that Denisovans gave modern humans a general biological advantage. The larger lesson is quieter. Extinction and inheritance are not opposites. A human population can stop existing as a recognizable group, while some of its biological ancestry remains distributed among descendants belonging to other populations. Denisovans therefore survive in a very limited but measurable sense inside human genomes, not as preserved people and not as a hidden race within modern humanity, but as fragments of population history. Ancient DNA made those fragments visible by giving researchers a reference point from the past. Without the genomes recovered from Denisovan remains, some unusual segments in living humans might have been far harder to interpret. With that comparison available, the forgotten lineage became connected to people living thousands of kilometers from Denisova Cave. The bones revealed that Denisovans existed. The genomes of later humans revealed that their story did not simply end when Denisovans disappeared from the visible world. Denisovan ancestry is not distributed evenly among living human populations, and that uneven pattern is one of the clearest signs that the history behind it was complex. Some populations in Oceania carry comparatively strong Denisovan ancestry signals, while many populations elsewhere carry much smaller amounts or patterns that appear to come from different Denisovan-related sources. This does not mean that any living population is simply closer to being Denisovan as a whole. Modern populations have their own long histories, and the archaic segments they carry represent only small parts of genomes shaped by countless generations of Homo sapiens ancestry. The differences begin with ancient contact. If separate groups of modern humans encountered different Denisovan-related populations at different places or times, they would not have received identical genetic material. Later, population movements then changed the picture again. Descendants migrated, separated, mixed with neighboring groups, expanded in number, or passed through periods when only a smaller number of people contributed to later generations. Each process could increase, reduce, or redistribute the Denisovan-derived segments that survived. Recombination also broke inherited stretches into smaller pieces every generation. Genetic drift, the random change in variant frequencies that occurs as populations reproduce through time, could preserve some segments and eliminate others without any biological advantage being involved. Natural selection added another layer. Some archaic variants were probably disadvantageous in the genetic background of Homo sapiens and became less common. Other Denisovan-derived variants appeared to have been useful under particular environmental conditions and increased in frequency. A well-studied example involves genetic variation associated with adaptation to low oxygen conditions among Tibetan populations, where a Denisovan-related version near the EPAS1 gene contributed to later adaptation at high altitude. This is an important example, but it should not be stretched into the claim that Denisovans broadly equipped modern humans for difficult environments. Each inherited variant has its own history, and most archaic DNA did not necessarily provide an advantage. The uneven distribution seen today is therefore the result of several processes layered on top of one another. Different encounters, different source populations, later migration, population mixing, random inheritance, and selection. The pattern also reaches back to the beginning of our wider human history. When Homo sapiens expanded across Asia, our ancestors were not one genetically uniform population moving through a uniform world. Different groups followed different routes and experienced different contacts. A lineage encountered by one population might never have been encountered by another. Even after Denisovan ancestry entered Homo sapiens, later movements could carry it far from the original place of contact. This is why researchers cannot look at a modern map, find the populations with the most Denisovan ancestry, and assume Denisovans lived in exactly those places. Present-day ancestry is the end product of a long demographic history rather than a frozen image of the ancient world. The uneven pattern is useful precisely because it preserves traces of that history. When scientists compare which Denisovan-derived segments are shared and how closely those segments resemble the known Altai-Denisovan genome, they can test models involving different episodes of introgression and different Denisovan-related populations. Some details remain debated as methods and data sets improve, so fixed counts of encounters should be treated cautiously. The broader conclusion is much stronger. Denisovan ancestry entered different human populations through a history that was neither single nor simple. What survives in living genomes is therefore not a uniform inheritance from one forgotten people. It is a fragmented record of different meetings, followed by tens of thousands of years of human movement and population change, leaving each modern population with a different genetic echo of the ancient world. That difference is not a ranking of human populations and does not divide living people into ancient biological categories. It is simply evidence that ancestry follows population history. The Denisovan contribution became uneven because the descendants who carried it did not remain together, and because every later generation reshaped which fragments survived and where they were found. Genes can sometimes act like imperfect maps of ancient movement, not because they record roads or destinations, but because ancestry changes as populations separate, migrate, and meet again. Denisovan-derived DNA provides a striking example. When researchers compare genetic segments carried by living populations, they can examine where those segments are found, how similar they are to the Denisovan genome recovered from Siberia, and whether different populations share the same inherited fragments. These patterns allow scientists to test possible histories of movement across Asia and Oceania, yet the word map must be used carefully. A genetic signal does not mark the exact place where an ancient encounter occurred. People continued moving after Denisovan ancestry entered Homo sapiens, and later populations mixed with one another many times. A segment found thousands of kilometers from Denisova Cave today may have traveled through generations of descendants before reaching its present distribution. The value of these genetic traces lies not in providing a fixed prehistoric geography, but in revealing connections that would otherwise remain invisible. If two modern populations share similar Denisovan-derived segments, their ancestors may share some history after archaic ancestry entered Homo sapiens. If the segments differ substantially, separate episodes of contact or different Denisovan-related source populations may offer a better explanation. Researchers compare such possibilities with demographic models, ancient genomes, archaeological evidence, and the known relationships among living populations. No single line of evidence is enough by itself. The genetic record is especially important in regions where ancient DNA preserves poorly, because warm and humid environments can destroy the molecules that might have survived in colder caves. In those landscapes, living genomes can preserve indirect evidence of ancient contact, even when the bodies of the people involved have almost entirely disappeared. This is one reason Denisovan ancestry has reshaped our understanding of the human expansion across Asia. Homo sapiens did not simply move eastward along one path and encounter one archaic population once. Different groups of our ancestors likely moved through different regions and became connected with different populations already living there. Some of those encounters left no known descendants. Others introduced ancestry that survived and later traveled with expanding human populations. By following those inherited fragments, scientists can sometimes identify broad directions of population history that archaeology alone cannot resolve. Still, genetics cannot reveal every root. Similar patterns can sometimes arise from different demographic histories, and later population mixing can blur earlier signals. Researchers therefore test competing explanations rather than treating one reconstruction as a final map. This uncertainty is especially important when asking where Denisovans themselves lived. The distribution of Denisovan ancestry in Oceania does not mean that Denisovans necessarily occupied every island where that ancestry is now found. Their genetic contribution could have entered ancestors elsewhere and then moved with later human migrations. In the same way, lower levels of Denisovan ancestry in other regions may reflect later dilution rather than a direct measure of how often ancient populations met there. The beginning of our own broader history is hidden inside these movements. Every migration changed not only where people lived, but also which pieces of older ancestry traveled with them. Some genetic fragments crossed mountains, coastlines, and islands because descendants carried them forward, while the populations that first contributed those fragments disappeared as distinct groups. Denisovan DNA, therefore, functions less like a prehistoric GPS coordinate and more like a trail of inherited connections. It can show that populations once touched, that ancestry moved beyond the place of contact, and that later generations carried pieces of vanished lineages into new environments. When combined with fossils and archaeology, those connections help transform the story of human origins from a sequence of isolated populations into a history of movement across space and time. The genes do not tell us what those travelers saw, what they feared, or what they called the people they met. They tell us something narrower, but more secure. The forgotten Denisovan lineage entered the ancestry of moving Homo sapiens populations, and those fragments were carried far beyond the places where the original encounters occurred. The name Denisovan sounds singular. It suggests one ancient people with one shared history, one homeland, and one clear place in the human family. Genetics has made that picture increasingly difficult to maintain. The genome recovered from Denisova Cave gave researchers a powerful reference point, but later comparisons with DNA from living humans revealed that not all Denisovan-derived ancestry looks equally close to that Siberian individual. Some inherited segments appear more closely related to the Altai Denisovan genome, while others seem to come from populations that had already been separated from that branch for a long period before contact with Homo sapiens occurred. This means that the word Denisovan may describe a broader lineage containing several populations rather than one uniform group spread across Asia. The distinction matters because population structure develops naturally when groups become separated by distance, environment, or demographic history. People living in different regions do not need to become different species for their genomes to begin following different paths. If contact between those regions remains limited over many generations, genetic differences can accumulate. Later movement can reconnect some groups, while others remain isolated. Denisovan-related populations may have experienced this kind of history across the enormous landscapes of eastern Eurasia. The individuals represented at Denisova Cave therefore cannot automatically stand for every population carrying Denisovan ancestry elsewhere. The same caution applies to the Denisovan ancestry found in living humans. Genetic studies support a history involving more than one Denisovan-related source contributing ancestry to Homo sapiens. Some models distinguish several introgression signals, but the exact number should not be treated as a final count of ancient populations. Models depend on assumptions about population size, separation, later migration, and the structure that existed before contact. Different demographic histories can sometimes create similar patterns in genomes. What remains important is the broader conclusion that a single encounter with one genetically uniform Denisovan population is unlikely to explain all of the surviving evidence. This brings the question of human origins back to geography. A population related to the Denisovans in Siberia may have shared deep ancestry with another Denisovan-related population much farther south or east, while also being genetically distinct from it. Homo sapiens moving through Asia could therefore have encountered different descendants of the same broader lineage in different places. Their ancestry could then enter separate modern human populations and later be redistributed by migration. This helps explain why the genetic legacy of Denisovans is uneven and why some Denisovan-derived segments in living humans do not match the Altai genome equally well. It also warns us against turning a scientific label into an imagined culture. The people grouped under the term Denisovan may never have thought of themselves as members of one population. Groups separated by large distances may have had different technologies, diets, social traditions, and ways of using the landscape, although those differences cannot be assumed without archaeological evidence. Genetics can show biological structure. It cannot automatically reconstruct social identity. At the beginning of our wider human story, that distinction becomes essential. Homo sapiens did not meet an abstract category called the Denisovans. Our ancestors encountered real populations, living in particular environments, each carrying its own demographic history. Some of those populations contributed ancestry to later humans, while others may have left no detectable descendants. The evidence surviving today is therefore a filtered record of only part of the ancient population network. The name Denisovan remains useful because it identifies a major lineage closely related to Neanderthals and recognized through molecular evidence. But the name should not flatten the diversity hidden inside it. Ancient DNA has moved the question from whether Denisovans existed to how many different population histories may be contained within that lineage. The forgotten humans revealed by one Siberian genome may turn out not to represent one lost people at all, but a broader family of related populations whose separate histories crossed Asia before some of them became part of the ancestry of Homo sapiens. When researchers compare Denisovan-derived DNA across living human populations, the signals do not all point back to exactly the same ancient source. Some inherited segments resemble the high-quality Denisovan genome from the Altai more closely than others do. That difference matters because it suggests that the people contributing Denisovan ancestry to Homo sapiens were themselves genetically structured. In other words, the lineage identified at Denisova Cave was probably only one branch within a broader population history. Genetic studies have found evidence consistent with several episodes of introgression from distinct Denisovan-related populations into modern humans. The exact number depends on the models and datasets being used, so it is safer to speak of multiple signals rather than to treat any fixed count as a final description of the past. Each signal represents a pattern that needs explanation, not a newly discovered species waiting for a name. If two Denisovan-related populations had been separated for many generations before meeting different groups of Homo sapiens, the fragments they contributed would carry different relationships to the Altai Denisovan genome. Later migrations could then move those fragments into new populations, where recombination would break them into smaller pieces, and genetic drift would alter how common they became. By the time researchers examine living genomes, they are therefore looking at the end of a very long chain of events, rather than at the original encounters themselves. This is why multiple signals of Denisovan ancestry have to be interpreted through population history. One pattern may fit contact with a group relatively close to the Siberian Denisovans, while another may require a source that had diverged from the Altai population much earlier. The differences can be informative, but they do not provide complete biographies of those source populations. We do not know their names, their social identities, or the precise boundaries of their territories. Genetics can estimate relationships among populations, but it cannot turn those relationships into lost cultures. This distinction becomes especially important when thinking about the beginning of the modern human story in Asia. Homo sapiens did not meet one timeless Denisovan population waiting across the entire continent. Different groups of our ancestors likely moved through different regions at different periods, encountering human populations that had already developed their own local histories. Some contacts left ancestry that survived. Other encounters may have left little or none that can still be detected. The fragments visible today are therefore a filtered record. They preserve only the portions of archaic ancestry that passed through all the later changes in human populations. Natural selection removed some inherited variants more efficiently than others. Chance eliminated many fragments. Population expansions increased the descendants of some ancestral groups, while later admixture diluted or redistributed earlier ancestry. A strong signal in a living population today is consequently not a simple measure of how many Denisovans once lived nearby. It reflects both the ancient contact and everything that happened afterward. This is one reason researchers compare many parts of the genome rather than relying on a single inherited segment. Shared patterns across numerous individuals can help distinguish between one source population and several sources, while comparisons with ancient genomes provide reference points for how closely related those sources may have been. Even then, alternative demographic models must be considered because population structure can sometimes imitate the genetic signature of a separate admixture event. The purpose of the analysis is not to multiply mysterious human groups whenever a pattern looks unusual. It is to find the simplest population history that explains the evidence without ignoring its complexity. Denisovans are therefore teaching us to be precise about the meaning of a forgotten lineage. The name identifies a real branch of ancient humanity, but the ancestry beneath that name may contain several histories that had already begun separating before Homo sapiens encountered them. What ancient DNA reveals is not a single hidden people preserved unchanged across Asia, but layers of relationship among populations whose paths separated, crossed again, and eventually became embedded in the ancestry of later humans. The more these signals are compared, the clearer it becomes that the question of who the Denisovans were cannot be answered by one genome alone. It requires understanding how many different population histories may be hiding inside the genetic traces that survived. A genetic difference can be real without marking the boundary of a separate species. This is especially important in the Denisovan story, because ancient genomes can reveal population structure more easily than they can resolve formal biological categories. When researchers detect Denisovan-related ancestry that differs from the Altai Denisovan genome, the first conclusion is not that another unknown human species has been discovered. A population can become genetically distinctive simply because it has been separated from related populations for many generations. Distance, Small population size, migration barriers, and random genetic drift can produce differences while groups remain part of a broader related lineage. Denisovans themselves provide a clear reason for caution. The first researchers who described their nuclear genome recognized a distinct population history closely related to Neanderthals, but deliberately avoided assigning a formal species or subspecies name. Later discoveries made the boundary question more complicated. Denisovans and Neanderthals were genetically distinguishable, yet they sometimes had children together. Both also exchanged ancestry with Homo sapiens. This does not mean that every ancient human population was biologically identical. It means that the categories used to describe human evolution do not always behave like sealed boxes. Fossils, genomes, geography, and reproductive contact preserve different aspects of the same history, and no single measure automatically settles where one species ends and another begins. The same restraint is necessary when several Denisovan ancestry signals appear in living humans. Genetic models may infer source populations deeply separated from the Altai Denisovans, but a source population inferred from DNA is not equivalent to a population known from securely identified anatomy, geography, chronology, and genetics. It is evidence that some ancestral population is needed to explain the genomes. Researchers then ask whether the signal reflects a distinct population, older population structure, repeated contact, or some combination of these processes. Recent work on human evolutionary models has emphasized that ignoring long-standing population structure can sometimes make admixture events appear more numerous or definite than they really were. That warning matters directly to forgotten human lineages. Ancient DNA is powerful, but it can create an illusion of precision if every unusual signal is immediately given a name and a story. The beginning of our own species was shaped by populations that divided and reconnected repeatedly, so genetic differences are expected even within related groups. A population living far from the Altai could become genetically distinct from Siberian Denisovans without becoming a separate species in any simple sense. If its descendants later contributed ancestry to Homo sapiens, modern genomes might preserve that difference, even though no fossil from the source population has been identified. This is why scientists separate questions that popular accounts often combine. Was there a genetically distinct ancestral population? Did it belong within the broader Denisovan lineage? Did it possess anatomical differences recognizable in fossils? Was reproductive isolation strong enough to justify separate classification? These questions require different evidence, and they may not receive the same answer. Denisovan anatomy also remains incompletely known, so there is still no reason to turn every genetic branch into a formal taxonomic unit. The more careful interpretation is more revealing. Denisovan may represent a broad, structured lineage whose populations became separated across Asia, accumulated differences, and later contributed ancestry to different Homo sapiens groups. Some of those populations may eventually prove distinctive enough to change scientific classification. Others may remain best understood as branches within a complex Denisovan population history. Human evolution was not organized according to the clean labels we apply afterward. Populations existed first. They moved, separated, met, and reproduced. Species names are scientific tools used later to describe patterns in that history. The forgotten lineage revealed in Siberia therefore becomes more interesting, not less, when we resist creating new species too quickly. Its genetic diversity may be telling us that one name hides several populations, while the evidence still asks us to leave open exactly how those populations should ultimately be classified. A ghost population is not a supernatural idea, and it is not a mysterious tribe invented to fill a gap in the past. In genetics, the phrase is used for an ancestral population whose existence is inferred from patterns in DNA, even when scientists do not yet have a securely identified ancient genome from that population itself. The population is called a ghost because its direct biological record is missing or incomplete, while its influence appears indirectly in the ancestry of later people. This concept became important as researchers realized that human evolution could not always be explained by comparing only the fossils and genomes already in hand. Sometimes a living population carries genetic variation that does not fit neatly into a model containing only known groups. The unexpected pattern may suggest that another ancestral population contributed DNA somewhere in the past. That does not immediately tell scientists who those people were. It does not reveal their appearance, language, territory, or cultural identity. It simply indicates that the ancestry of known populations may include input from a source that has not yet been directly sampled. Denisovans once occupied a similar position in our understanding, although their case quickly became much stronger because researchers recovered ancient DNA from actual remains. Before that genetic identification, no complete Denisovan skeleton stood in a museum waiting to be recognized. Their discovery showed that a human lineage could remain almost invisible anatomically until molecular evidence brought it into view. Ghost populations take that principle one step further. Instead of beginning with a bone that yields DNA, researchers may begin with the genomes of other populations and notice that the relationships among them cannot be explained without an additional source of ancestry. Statistical models can then test whether gene flow from an unsampled population provides a better explanation. Yet these models must be treated cautiously. A genetic pattern that appears to require an unknown population can sometimes be produced by long-lasting population structure within a known ancestral group. Imagine an ancient population already divided into regional communities that exchange genes only occasionally. If researchers later model that ancestral population as though it had been completely uniform, the surviving genetic differences might resemble evidence of admixture from a separate, hidden group. The signal can therefore be real, while the simplest interpretation remains uncertain. This problem reaches directly back to the beginning of Homo sapiens. Human populations in Africa and Eurasia were rarely perfectly isolated or perfectly uniform. Groups separated, expanded, contracted, and sometimes reconnected. Deep population structure can survive in genomes for very long periods, creating patterns that later resemble contributions from distinct lineages. Scientists therefore compare different demographic models rather than announcing a new human population whenever an unusual sequence appears. Fossils, archaeology, ancient proteins, chronology, and ancient DNA can all help test whether a ghost population eventually becomes connected to a known group. Until that happens, the responsible language remains provisional. Researchers may describe an unsampled archaic population, an unknown ancestral source, or a ghost lineage, while avoiding claims that go beyond the evidence. The Denisovan story provides a useful lesson here because it shows both the power and the limits of genetic inference. Once a high-quality ancient genome existed, researchers could compare Denisovan ancestry directly with DNA in living humans. But when some inherited segments did not match the Altai-Denisovan equally well, the possibility arose that additional Denisovan-related populations had contributed ancestry. Those populations are not automatically new species. They may represent branches within the broader Denisovan lineage that remain unsampled. In other cases across human evolutionary research, ghost signals may reflect populations for which no securely identified fossil has yet been found. The important point is not to fill every gap with an imagined people. The important point is that the human past contained more population structure than the surviving fossil record can directly display. At the origin of our wider human story, many groups may have left descendants without leaving a recoverable genome of their own. Their existence can sometimes be approached through the patterns they left inside others, but those patterns must be interpreted with restraint. A ghost population is therefore not a hidden civilization waiting to be uncovered. It is a scientific placeholder for ancestry that appears to be present even though its source remains incompletely known. The concept reminds us that the absence of a fossil or genome is not proof that no population existed, while also reminding us that genetic models are not the same as direct evidence. Somewhere between those two truths lies one of the most difficult questions in human origins. How many ancestral populations shaped us without leaving enough of themselves behind to be recognized clearly? A genetic signal can reveal that something happened in the ancestry of a population, but it is not the same kind of evidence as a fossil lying in a dated archaeological layer. This distinction is essential when researchers study possible ghost populations. Genomes preserve patterns created by inheritance, separation, migration, population size, and interbreeding. When one pattern cannot be explained easily by the populations already included in a model, scientists may test whether ancestry from an unsampled group improves the explanation. That possibility can be powerful, but it remains an inference about population history rather than a recovered individual. A fossil gives researchers a physical specimen that can be examined for anatomy, context, age, and sometimes molecules. A genetic signal detected indirectly in other people provides a different kind of information. It may indicate that an ancestral source existed, yet it does not automatically reveal where that population lived, what its members looked like, or whether it should be classified as a distinct species. Models are built from assumptions about how populations were structured, when they separated, and whether they exchanged ancestry. If those assumptions are too simple, the model can interpret an old pattern of population structure as though it came from a separate episode of admixture. Imagine an ancestral human population spread across a large region, with communities connected only occasionally. Genetic differences could develop between those communities, even though they belong to a broader connected population system. If a later analysis treats that ancient population as completely uniform, surviving differences may resemble ancestry introduced by an unknown external group. The signal would still reflect genuine history, but the explanation could be wrong. Recent research has shown how seriously this problem must be taken by demonstrating that models ignoring population structure can infer admixture even from simulated histories in which no such event occurred. This does not erase the strong evidence for established contacts involving Neanderthals, Denisovans, and Homo sapiens. It shows why proposed events need to be tested against alternative demographic histories. The issue becomes especially important when looking toward the beginning of Homo sapiens. Human populations in Africa and Eurasia were not necessarily isolated units with simple boundaries. Communities could remain connected at low levels across long periods, divide into regional populations, reconnect, and later contribute unequally to descendants. Those processes can leave deep genetic structure without producing a neat fossil category for every branch. The absence of a fossil, therefore, does not invalidate a genetic inference, but neither does an unusual signal create a fossil population by itself. Direct ancient DNA can sometimes transform the situation. Before Denisovans were recognized, their lineage was absent from the conventional picture built from recognizable skeletons. Once DNA was recovered from actual remains in Denisova Cave, researchers had a physical reference connected to a genome. Later fossils and proteins could then be compared with that reference. A ghost population lacks that direct anchor. Until a securely identified specimen or ancient genome is found, its biological identity remains less certain. This difference protects the study of human origins from a common mistake, turning statistical necessity into a fully imagined people. A model may require ancestry from an unsampled source, but it cannot supply that source with a face, a language, a homeland, or a culture. Those details must come from independent evidence. Even the relationship between an inferred source and known lineages can change when new genomes become available, or when population structure is modeled more realistically. Scientists therefore compare models and look for agreement between genetics, fossils, archaeology, chronology, and molecular evidence. Ghost signals are valuable because they tell us where the current picture may be incomplete, but they are also reminders of restraint. Ancient DNA has taught us that forgotten lineages can be real, as the Denisovan discovery proved. But not every unexplained genetic pattern has already revealed another Denisovan. Some signals may eventually lead to newly sampled populations. Others may be better explained by deep structure among populations already connected within the human family. Until additional evidence separates those possibilities, the genome can point toward missing ancestry without giving us the missing people themselves. Ancient DNA has transformed the study of human origins because it can reveal relationships that fossils alone may never expose. But genetic reconstruction has limits that become more important as the questions grow deeper. A genome is not a complete record of a population. It is one biological sample shaped by ancestry, recombination, mutation, natural selection, genetic drift, and the demographic history of many earlier generations. When researchers compare ancient and modern genomes, they build models that attempt to explain observed patterns through population splits, migration, changes in population size, and episodes of gene flow. These models are powerful because they can test whether one historical scenario fits the data better than another, yet they are not direct recordings of what happened. Different population histories can sometimes leave similar genetic patterns. A signal interpreted as admixture from an unknown population may, under another model, be explained through deep structure within an ancestral population that was never completely uniform. A period of isolation may appear longer or shorter depending on which genomes are available for comparison. A population that seems absent may simply remain unsampled because its remains have not survived or because ancient DNA has not been recovered from the regions where it lived. This is why genetic reconstruction works best when evidence from fossils, archaeology, ancient proteins, geography, and chronology can be brought into the same picture. Denisovans illustrate both the strength and the restraint of this approach. Their existence is supported directly by ancient DNA from identified remains, and later molecular evidence has expanded their known geographic range. Yet even with those discoveries, researchers cannot reconstruct every Denisovan population or every encounter they had with Homo sapiens. The genomes of living people contain several Denisovan-related signals, but those signals do not reveal the names, cultures, or exact territories of the source populations. They show relationships among ancestors, not the social worlds in which those ancestors lived. This boundary reaches back to the beginning of our own story. Human populations were probably structured across space, with groups separating and reconnecting over long periods. If only a small fraction of those populations left recoverable genomes, then any reconstruction must be based on an incomplete sample of a much larger network. Adding another ancient genome can therefore change the model, because a population previously represented only indirectly may suddenly become visible. The Denisovan discovery itself is a reminder of how dramatic that change can be. Before their DNA was recovered, the lineage was not recognized as a major lineage within the human family. Afterward, genetic fragments in living people that had lacked a clear reference could be connected to a real ancient population. Future discoveries may do something similar for other unsampled lineages, but science cannot assume in advance what those discoveries will reveal. A statistical ghost must remain a hypothesis about ancestry until direct evidence strengthens its identity. Even then, classification may remain uncertain because human populations repeatedly exchange genes across boundaries that were never absolute. Genetic reconstruction, therefore, does not produce a final family tree carved permanently into the past. It produces the best supported explanation available from the evidence currently preserved. New fossils can alter it. New genomes can reveal previously hidden relationships. Better models can show that an older interpretation was too simple. This is not a failure of the method. It is the normal process of reconstructing a history from fragments. The most reliable conclusion is also the most modest one. Our ancestry contains traces of populations that are not yet fully represented in the fossil or genomic record. But the existence of an unexplained signal does not justify inventing a complete people around it. Ancient DNA can show that the human past was more diverse and interconnected than once imagined, while still leaving some identities unresolved. The forgotten lineage at the center of this story became visible because evidence eventually crossed the boundary from inference to direct molecular identification. Other lineages may still remain on the far side of that boundary, detectable only as patterns whose true sources have not yet been found. Extinction in the human past does not always arrive as a single dramatic event. A population can disappear without one catastrophe, one battle, or one moment that archaeology can identify as the end. Denisovans are a good example of why this matters. They are no longer present as a distinct human population, yet the evidence does not point to one securely established cause that explains their disappearance across all of Asia. Their known history suggests that Denisovan-related groups were spread across different regions, lived under different environmental conditions, and were genetically structured. A population living in the Altai could therefore have faced a different demographic future from a related population on the Tibetan Plateau or farther east. Even within one region, disappearance can happen gradually. Small populations are especially vulnerable to changes in birth rates, deaths, migration, and the loss of genetic diversity. If neighboring groups become more numerous, a smaller population may shrink without being destroyed suddenly. Individuals can move into other communities, descendants can inherit ancestry from both populations, and over generations the older population can cease to exist as a clearly separate group. This possibility matters for Denisovans because their DNA entered some Homo sapiens populations. Their ancestry survived, even though Denisovans themselves eventually disappeared as an identifiable population. That survival argues against imagining extinction only as complete biological erasure. Some communities may have declined, some individuals may have joined other populations, and some Denisovan ancestry may have continued through descendants. The relative importance of these processes remains uncertain. The archaeological record is too incomplete to reconstruct one final sequence for the entire lineage. Recent work at Denisova Cave has refined the chronology of human occupation there and suggests that environmental change alone does not provide a simple explanation for the disappearance of Denisovans from the Altai. Researchers have considered demographic factors, competition for resources, and encounters with expanding Homo sapiens among possible influences, but none should be treated as a proven universal cause. Conditions elsewhere in Asia could have been different. Climate changed repeatedly during the long history of Denisovan-related populations, but climate change does not automatically produce extinction. Human groups can move, change their ranges, alter how they use resources, or become connected with neighboring populations. The outcome depends on population size, geography, available habitats, and contact with other people. This returns us to the beginning of our wider human history. When Homo sapiens expanded through Eurasia, our ancestors entered landscapes that already contained other human lineages. Contact could have introduced competition in some places, but it also produced interbreeding. These possibilities are not mutually exclusive, and neither needs to have operated in the same way everywhere. A Denisovan-related community could decline in one region while another persisted elsewhere. A local population might disappear from a cave while related groups continued beyond the area preserved in the archaeological record. The last known trace at one site is therefore not necessarily the last Denisovan anywhere. Scientists must separate the disappearance of evidence from the disappearance of a lineage. Fossils can stop appearing because populations vanish, but also because preservation fails, occupation shifts, or later sites have not yet been found. Ancient DNA has repeatedly shown that the record contains gaps large enough to hide important populations. For Denisovans, those gaps remain substantial. What can be said safely is that their independent population histories eventually ended, while fragments of their ancestry did not. There is no need to invent a sudden final disaster to explain that outcome. Human populations can fade through demographic decline, movement, repeated mixing, local replacement, or combinations of these processes unfolding across many generations. The forgotten lineage at the center of this story may therefore have disappeared not through one recognizable ending, but through many local endings that left different traces. Some ancestry was lost, some was carried forward. The absence of a single dramatic cause is not an empty answer. It reminds us that evolution can change populations quietly until a lineage that once occupied real landscapes can no longer be found as a separate people. Climate, geography, and population size can all influence whether a human population persists, but none of them works as a simple switch that turns a lineage off. Denisovan-related populations lived across environments that differed greatly from one another, from the Altai region to the high Tibetan Plateau, and probably across broader parts of eastern Eurasia. In each place, climate could change the availability of plants, animals, water, and habitable territory. Colder or drier periods might reduce some resources while opening others, and warmer phases could alter migration routes and ecosystems again. Yet, climate change alone does not explain why Denisovans disappeared. Humans can respond to environmental change by moving, changing how they use landscapes, or becoming connected with neighboring populations. The archaeological record at Denisova Cave itself does not show a simple moment when the environment collapsed and Denisovans immediately vanished. This is important because a neat environmental catastrophe would be easier to tell as a story, but the evidence does not require one. Geography may have mattered in a quieter way. Mountains, plateaus, deserts, forests, and coastlines can separate populations, reducing contact between communities that share deeper ancestry. If Denisovan-related groups were distributed across large parts of Asia, some populations may have remained isolated from one another for long periods. Isolation can allow genetic differences to accumulate, but it can also make small communities more vulnerable if their numbers fall. A population spread thinly across difficult terrain may have fewer opportunities to exchange mates with neighboring groups, and random demographic events can then have larger effects. This does not mean that Denisovans everywhere were small. Genetic evidence suggests complicated population histories, and the true sizes of many Denisovan-related groups remain uncertain. What matters is the principle that population size changes the way chance operates. In a smaller population, a run of poor reproductive years, local resource shortages, disease, migration, or the loss of a few family lines can have consequences that would be easier for a larger population to absorb. Over many generations, these pressures can reduce diversity and make recovery more difficult. The arrival and expansion of Homo sapiens may have added another influence, but here too the evidence should not be forced into a simple replacement story. Modern humans could compete with other populations for some resources, yet they also interbred with Denisovan-related people. Local outcomes may therefore have differed. In one region, contact might have contributed to demographic absorption. In another, Denisovan-related populations may have continued for a longer period. Elsewhere, changing environments or geographic isolation may have been more important than contact with Homo sapiens. This brings us back to the beginning of our own human story. The ancestry of living people was shaped not only by populations that expanded successfully, but also by populations that became smaller, fragmented, or merged into others. Denisovans show how these processes can overlap. A population can lose its separate demographic identity, while some of its genes persist in descendants. Climate can change the landscape without causing immediate extinction. Geography can protect a group from contact for a time, while also limiting connections. Small population size can increase vulnerability without guaranteeing disappearance. None of these factors needs to act alone. The end of Denisovan population histories may have unfolded differently across Asia, with local declines, migrations, admixture, and environmental pressures occurring at different times. The last Denisovan trace at any one site should therefore not be confused with the last Denisovan population anywhere, because preservation and discovery remain uneven across the continent. What ancient DNA gives us is not a single cause, but evidence that the vanished lineage was connected to populations whose histories were already diverse. Their disappearance may therefore be better understood as a long demographic transformation rather than one event, a gradual narrowing of separate Denisovan populations until the lineage survived most clearly not as an independent people, but as fragments of ancestry carried forward inside others. When we say that the Denisovans vanished, the word can create a picture that is cleaner than the evidence allows. It sounds as though a distinct people existed, reached a final generation, and then disappeared completely. Population history rarely works so neatly. The Denisovans are no longer known as an independent human population, but parts of their ancestry remain in living people. That means disappearance happened at more than one level. Their communities, identities, and separate demographic histories eventually ended, while some genetic material continued through descendants who belonged to later Homo sapiens populations. In this sense, a lineage can cease to exist without every biological trace being erased. The distinction matters because interbreeding had already connected Denisovan-related populations with Homo sapiens before Denisovans disappeared from the visible record. When descendants inherited ancestry from both populations, those Denisovan-derived segments entered a much larger human population network. Recombination then broke them into smaller pieces across generations. Many fragments were lost, some remained through chance, and a limited number appear to have been favored under particular environmental conditions. The result is not a hidden Denisovan population surviving inside modern humanity. It is ancestry from an ancient population surviving inside people whose overall histories are overwhelmingly those of later Homo sapiens populations. This is similar to the way a river can lose its separate channel after joining another river while some of its water continues downstream, although human ancestry is far more complicated than that image suggests. The genetic record allows us to recognize that contribution, but it cannot tell us exactly how the demographic disappearance unfolded in every region. Denisovan-related populations were probably structured across a wide geographic range, and their endings may not have occurred at the same time. A group in one area could have declined, while another persisted elsewhere. Some communities may have become absorbed into larger populations through repeated interbreeding. Others may have disappeared locally without leaving descendants that can still be detected. Because the fossil record is sparse, the last known Denisovan evidence from a particular site cannot safely be treated as the final Denisovan presence anywhere in Asia. What appears to be an ending in archaeology may sometimes be a gap in preservation or discovery. This uncertainty brings us back to the beginning of our own wider history. Homo sapiens became the only surviving human lineage. But becoming the only surviving lineage did not require every older population to leave no contribution behind. Neanderthal and Denisovan ancestry both demonstrate that population replacement and population mixture can occur within the same broad history. A group may lose its separate identity, while some descendants become part of another expanding population. From an evolutionary perspective, that makes the boundary between survival and extinction less simple than everyday language suggests. Genes can survive after a population does not, but genes alone are not the population. A Denisovan-derived segment does not preserve a community, a language, a memory, or a way of life. Those aspects of Denisovan existence may have vanished almost completely. What remains is a biological connection, measurable but fragmentary, between people living today and populations that once occupied ancient Eurasia. This is why it is more accurate to say that Denisovans disappeared as distinct populations rather than imagining that they were erased in every possible sense. Their ancestry passed into some Homo sapiens groups, was reshaped by later demographic history, and became scattered across descendants far removed from the original encounters. The forgotten lineage, therefore, has two endings at once. As a separate population, it is gone. As a contributor to later human ancestry, it is not entirely absent. That paradox is one of the most important lessons ancient DNA has given us, because it changes what we mean when we ask whether an ancient human lineage truly vanished. The people themselves cannot be recovered from those inherited fragments, yet the fragments prevent us from treating their disappearance as total biological erasure. They show that the history of one human population can continue inside another after names, territories, and separate communities have gone. The question is therefore not simply whether Denisovans survived, but what kind of survival ancestry can represent. Human evolution is often drawn as a procession, with one form appearing after another until modern humans stand alone at the end. That image is easy to understand, but ancient DNA has shown that it is too simple. The deeper past contained several human populations living at the same time, sometimes separated by enormous distances and sometimes close enough to meet. Neanderthals, Denisovans, and Homo sapiens were not steps on a ladder leading toward one predetermined result. They were related populations with their own histories, shaped by movement, isolation, changing environments, and contact with others. The Denisovan discovery made this especially difficult to ignore, because their existence was first revealed through ancestry rather than through a complete anatomical portrait. Once their genome could be compared with Neanderthals and modern humans, the relationships among these populations no longer looked like a sequence of clean replacements. Denisovans and Neanderthals shared a relatively recent common ancestry with each other, yet their populations later diverged. After that separation, contact did not disappear forever. Genetic evidence shows that Denisovans and Neanderthals exchanged ancestry, and later Homo sapiens also received ancestry from both lineages. The result is a history in which branches separate and then cross again. This does not erase the differences between ancient populations. Long periods of limited contact created real genetic structure, and fossils show that different human groups could develop distinctive anatomical patterns. But those differences existed within a world where reproductive boundaries were sometimes porous. A population could remain genetically recognizable while still contributing ancestry to another population. This is why modern human origins cannot be described simply as the victory of one lineage over all others. Homo sapiens eventually became the only surviving human population, but the genomes of living people show that some earlier lineages were not erased without contribution. Their ancestry was absorbed unevenly into expanding human populations and reshaped over generations. Denisovan-derived DNA found today in populations across Asia and Oceania is one result. Neanderthal-derived ancestry provides another. These inherited fragments do not make living humans members of those ancient populations, and they do not preserve the cultures or identities of the people who contributed them. They preserve biological relationships. Population structure existed within Africa, migration connected and separated communities, and later movements beyond Africa brought some Homo sapiens populations into contact with humans whose ancestors had followed different paths. Ancient DNA has made these connections measurable, but it has also shown why a single tree can be misleading. A tree is useful for representing shared ancestry and divergence. It is less successful at showing what happens when separated populations meet again and exchange genes. Human evolution, therefore, begins to resemble a network laid over a branching history. Some connections were strong enough to leave descendants. Others may have disappeared without detectable genetic traces. Many populations remain poorly sampled, and some may never be represented by recoverable DNA. Denisovans remind us of that missing diversity because science recognized an important human lineage from remarkably little physical material. Their discovery suggests caution whenever the fossil record looks empty. Empty space in a museum display does not necessarily mean empty space in the ancient world. Preservation is selective, excavation is incomplete, and molecular evidence survives under only some conditions. The history we can reconstruct is therefore a surviving fraction of a larger population landscape. Seen from this perspective, human evolution was never a march toward us. There is no evidence that Denisovans or Neanderthals were waiting to be replaced, and no scientific reason to describe Homo sapiens as the inevitable destination of earlier evolution. Different populations persisted for different lengths of time, encountered different environments, and left different kinds of descendants. Only one human lineage remains as a distinct population today, but that outcome does not turn the earlier world into a staircase. Ancient DNA instead reveals branching paths, crossings, disappearances, and inheritances, in which the forgotten Denisovans were one real population history among several that helped shape the ancestry carried forward by later humans. Ancient DNA can answer questions that once seemed unreachable, but its power becomes most useful when its limits are understood just as clearly as its strengths. A genome can reveal biological relationships between individuals and populations. It can show that two groups shared common ancestry, that populations separated for long periods, or that genetic material moved from one group into another through interbreeding. It can sometimes help estimate the order in which these events occurred, and test whether a simple population history is sufficient to explain the data. In the case of Denisovans, these methods transformed a tiny fragment of bone into evidence for a major human lineage, and later connected that lineage with ancestry carried by living populations across parts of Asia and Oceania. That is an extraordinary reach, but it is not unlimited. DNA does not preserve a spoken language. It does not contain a community's stories, rules, beliefs, memories, or sense of identity in a form that can simply be read back. A genome cannot tell us what an individual thought while looking across the Altai Mountains, how a Denisovan family understood kinship, or what meanings they gave to animals, caves, seasons, or death. Those questions require archaeological evidence of behavior, and even archaeology often leaves them only partially answered. Genetics is strongest when it is used for the kinds of questions that inheritance can actually address. By comparing genomes, researchers can reconstruct patterns of relatedness, detect some episodes of gene flow, and examine how populations changed through time. Yet even these conclusions depend on the samples and models available. Ancient DNA survives unevenly. Cold caves may preserve genetic material that disappears rapidly in warm and humid environments, creating a geographic bias in what can be recovered. A well-preserved genome from Siberia, therefore, does not mean that Siberia was the center of a lineage's entire history. It means that one person happened to leave material that survived long enough to be studied. Missing genomes from other regions may represent missing preservation rather than missing populations. The beginning of our own wider human history must be reconstructed through this uneven archive. Homo sapiens, Neanderthals, Denisovans, and other ancestral populations did not organize themselves according to the clean categories scientists use today. They moved, divided, met, and sometimes exchanged ancestry. Genetic models try to recover these processes from patterns left behind, but a model is an explanation of evidence rather than a recording of the past. Different demographic histories can sometimes create similar genetic signatures. If ancient population structure is simplified too much, researchers may infer an episode of admixture where long-standing connections among regional populations offer another explanation. This is why conclusions become stronger when genomes agree with fossils, proteins, archaeology, stratigraphy, and geography. Denisovans provide a particularly clear lesson. Their genetic identity is secure because ancient DNA came directly from identified remains, while later molecular evidence connected additional fossils to the broader lineage. Even with that foundation, however, the label Denisovan does not give us a complete portrait. We cannot assume that Denisovan-related populations across Asia looked identical, used the same technologies, followed the same social traditions, or considered themselves members of one people. Genetic differences suggest internal population structure, but they do not translate automatically into cultural boundaries. The same restraint applies to inherited Denisovan variants in living humans. A Denisovan-derived segment can show that ancient ancestry survived. It cannot tell us the personal circumstances under which that ancestry first entered Homo sapiens. It cannot reveal whether the individuals involved spoke mutually understandable languages, whether contact was brief or sustained, or what social relationship existed between their communities. Those details are lost unless another form of evidence can recover them. Understanding this boundary does not make ancient DNA less impressive. It makes the knowledge more reliable. The strongest lesson is not that genomes allow us to reconstruct every lost detail, but that they can recover certain relationships with remarkable precision while leaving other questions honestly open. Denisovans became visible because science learned how to ask molecules the questions molecules could answer. Their story also teaches us when not to ask those molecules for memories they never preserved. The past becomes more truthful when evidence is allowed to remain incomplete, because uncertainty is not an empty space to fill with invention. It is a boundary showing exactly where knowledge ends and where the unknown still begins. The Denisovans were forgotten not because they were insignificant, but because almost everything that once made them recognizable as a people disappeared. Their communities are gone. Their languages, social identities, and memories were not preserved in forms we can recover. For most of modern history, we did not even know that this branch of humanity had existed. Then, ancient DNA revealed that a tiny fragment of bone could contain a history larger than the fragment itself. From that beginning, the Denisovan story expanded outward. Genetic evidence placed them close to Neanderthals, while showing that they followed a distinct population history. Fossils and proteins connected Denisovan-related people with regions far beyond the Siberian cave where they were first identified. Traces in living genomes showed that some of them encountered Homo sapiens and had descendants whose ancestry continued through later generations. None of these discoveries gives us a complete Denisovan world, but together they answer the central question more carefully than any imagined reconstruction could. Denisovans were not a mysterious civilization erased from history, and they were not one simple population frozen in time. They were a branch of ancient humanity, probably composed of multiple related populations spread across parts of Asia, whose histories included separation, movement, contact, and interbreeding with other humans. What ancient DNA revealed was therefore not merely another name to add beside Neanderthals and Homo sapiens. It revealed how incomplete our earlier picture of human origins had been. The beginning of our own story did not take place in a world containing only one human lineage moving toward the future. It unfolded among several populations whose paths crossed in ways that fossils alone could not show. Some disappeared without contributing detectable ancestry to people living today. Others, including Denisovan-related populations, became part of later human ancestry through descendants. This does not mean that Denisovans survive secretly inside us. Genes are not people. A genetic fragment cannot preserve a voice, a relationship, a memory, or a culture. What it preserves is connection. That distinction may be the most important educational lesson of the entire story. Human history is not only the history of populations that remained visible, it is also the history of populations that disappeared, merged, fragmented, or became absorbed into others. Evolution does not preserve identities because they are important to us. It preserves whatever happens to survive through reproduction, environment, chance, and population change. The Denisovans remind us that scientific knowledge grows by accepting these limits rather than filling them with dramatic invention. We know that they existed because ancient molecules and securely identified remains support that conclusion. We know that they were related to Neanderthals, and that some Denisovan-related populations contributed ancestry to Homo sapiens. We know that their geographic history extended beyond Denisova Cave. But we still do not know the full range of their populations, the exact circumstances of many encounters, or how different Denisovan communities may have been from one another. Those unanswered questions are not failures. They are part of an honest reconstruction of the past. Every new fossil, protein sequence, ancient genome, or archaeological site has the potential to change the boundaries of what we understand, just as one small finger bone once changed the human family tree. The deeper value of that discovery is therefore not that science uncovered a lost people completely. It is that science learned how much could remain hidden even after a lineage had nearly disappeared from the visible record. Our species became the only surviving human lineage, yet our ancestry still carries evidence that we were never alone in the deeper past. Some branches ended. Some met us before they ended. Some left fragments that traveled farther through time than their own populations did. And somewhere beyond the fossils already found and the genomes already sequenced, there may still be traces of human populations whose existence has not yet become clear. If a forgotten lineage could remain hidden for so long and still survive as ancestry inside later humans, how many other human histories may be present in us, or beneath the ground, without yet having a name?